alam yang berlanjut, serta pengembangan pertanian yang berbasis pada kekayaan sumber daya tropika. Untuk mewujudkan visi tersebut, misi kami adalah menyelenggarakan proses pendidikan berstandar internasional di bidang pertanian berlanjut untuk menghasilkan lulusan yang profesional, kompeten, dan beretika. Di samping itu, kami melakukan pengembangan dan penyebarluasan iptek di bidang pertanian berlanjut, serta pemanfaatannya untuk mendukung pengembangan pertanian yang berbasis pada kekayaan sumber daya tropika. Di jurusan budidaya pertanian terdiri dari program studi S1 Agroekoteknologi dan S2 Ilmu Agronomi. Kedua program tersebut telah memiliki akreditasi A. Hal ini menjadi salah satu faktor peningkatan jumlah mahasiswa program studi S1 Agroekoteknologi dan S2 Agronomi selama 3 tahun terakhir. Jurusan budidaya pertanian ini memiliki 4 minat yang digolongkan berdasarkan laboratorium pendukung kegiatan penelitian dan pengabdian kepada masyarakat, yaitu laboratorium sumber daya lingkungan, laboratorium fisiologi tanaman, laboratorium pemuliaan tanaman, dan laboratorium klimatologi. Proses pembelajaran di jurusan budidaya pertanian terdiri dari kuliah offline dan online pada kondisi tertentu, tutorial, praktikum di laboratorium, dan praktikum di lapangan. Dalam 3 tahun terakhir, 484 jurnal hasil penelitian dosen jurusan budidaya pertanian telah dipublikasikan dan diterbitkan pada jurnal nasional maupun internasional. Sedang dalam 2 tahun terakhir telah menghasilkan 13 paten dan 20 buku.
Laboratorium Sumber Daya Lingkungan berperan dalam manajemen sumber daya lingkungan lahan marginal melalui pendekatan ekologi pertanian yang terbagi dalam beberapa kajian, yaitu urban farming, teknologi produksi tanaman, cekaman biotik dan abiotik, budidaya tanaman tanpa tanah, penataan landscape dan taman. Fisiologi tanaman adalah laboratorium yang dirancang untuk mengeksplorasi lebih jauh proses kehidupan tanaman, fungsi fisik, kimia, dan biologis dari kehidupan tanaman, mulai dari seluruh tanaman hingga tingkat sel. Hal ini sangat berhubungan dengan proses untuk menghasilkan organ dari sel tunggal atau sigot melalui embriogenesis, pembelahan dan diferensiasi sel, dan hormon pertumbuhan tanaman. Proses untuk menghasilkan biomasa melalui fotosintesis dan metabolisme, dan kemampuan beradaptasi dalam metabolisme primer dan sekunder untuk mengatasi kondisi stres. Laboratorium Pemuliaan Tanaman berperan dalam pendidikan mahasiswa agar kompeten di bidang pemuliaan tanaman dan layanan masyarakat melalui perpaduan integral antara ilmu dan seni yang terkait dengan ilmu pemuliaan tanaman seperti genetika, bioteknologi, dan teknik persilangan. Laboratorium Klimatologi mendukung pertanian di bidang iklim dengan menganalisis agroklimat dan data informasi iklim, manipulasi dan mitigasi iklim makro dan mikro, dan memprediksi keberhasilan tanaman dengan kesesuaian lingkungan tanaman.
Nama saya Syakila Putri, saya dari jurusan Budidaya Pertanian. Jurusan Budidaya Pertanian membekali kami tentang cara-cara peningkatan produksi tanaman, perbaikan teknik budidaya, genetik tanaman, hingga pengelolaan lingkungan tanaman. Mahasiswa diberi beberapa tugas dan mengikuti ujian secara terstruktur dan sistematis, sehingga kami sebagai mahasiswa dapat memahami kajian-kajian tersebut secara utuh, terintegrasi, dan berkualitas. Dosen budidaya pertanian sebagai tenaga pengajar memberikan materi berdasarkan textbook dan juga penelitian mutakhir dengan pengetahuan yang mendalam. Selain itu, dosen sekaligus mendidik moral dan etika mahasiswa sebagai calon sarjana dan juga anggota masyarakat.
Himpunan mahasiswa jurusan budidaya pertanian yang bernama Himadata adalah bagian dari lembaga kedaulatan mahasiswa yang merupakan representasi mahasiswa jurusan budidaya pertanian di Fakultas Pertanian UB. Mahasiswa jurusan budidaya pertanian memiliki kesempatan mendapatkan wawasan iptek dari dosen dan peneliti maupun praktisi dari luar negeri melalui program 3-in-1. Mahasiswa jurusan budidaya pertanian tidak hanya berprestasi di bidang akademik, tetapi juga di bidang non-akademik, baik dalam bentuk karya atau kreativitas ilmiah, maupun dalam pengembangan bakat minat berupa olahraga dan seni.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Welcome to the international webinar Crops Toward Environmental Sustainability as a series of Agronomy Conference Series, or AMYCOS. This webinar is held by Department of Agronomy, Faculty of Agriculture, Universitas Brawijaya. First of all, I would like to introduce myself. My name is Nita, as Master of Ceremony today. The Honorable Dr. Nurrahmi Ardiarini as Head of Department of Agronomy, the Honorable Dr. Afifuddin Latif Adirejo as Secretary of Department of Agronomy, the Honorable our speaker today, Mr. Muhammad Roviq, Master in Plant Science, Mr. Khairil Mahmud, Ph.D., and Mr. Raphael Perez, Ph.D. The Honorable our moderator, Mrs. Siska Fajriani, Master in Plant Science, Mr. Adi Setiawan, Ph.D., and Ms. Azeri Gautama Arifin, Master in Agriculture. And also the Honorable all participants. Ladies and gentlemen, in the beginning of this event, let us pray and praise to God because of His blessing and mercy, we can come together here with healthy condition. Before we come to the main session, let us start this by praying first, so the event that we will hold on this day will run well without any obstacle at all. Let's pray. Pray based on individual belief. Pray can begin. Done? So, ladies and gentlemen, on this nice occasion, let me deliver the structure of the event today as follows. First is opening, then second is opening speech from Dr. Nurrahmi Ardiarini as Head of Department of Agronomy. And then the first session will be delivered by Mr. Muhammad Roviq, Master in Plant Science, and followed by the second session that will be delivered by Mr. Khairil Mahmud, Ph.D. And at the third session will be delivered by Mr. Raphael Perez, Ph.D. In each session will be followed by question-answer session and the awarding of certificate session for keynote speaker. And in the last, there will be closing speech from Dr. Afifuddin Latif Adirejo as Secretary of Department of Agronomy. Ladies and gentlemen, now we invite Dr. Nurrahmi Ardiarini to give an opening speech. To Dr. Nurrahmi Ardiarini, time and place are yours.
Thank you, Ibu Paramita. The Honorable, the Dean of Agriculture Faculty, Brawijaya University, Honorable, the Vice of Dean, Agricultural Faculty, Honorable, the Head of Department of Agricultural Faculty, Honorable the speakers, dear the colleagues, participants, and the students. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. I'm pleased to welcome you all the first international webinar of AMYCOS, or Agronomy Conference Series, organized by the Department of Agronomy, Faculty of Agriculture, Brawijaya University. And I am also grateful to all of you for being part of this webinar. And I would like to take this opportunity to extend a warm welcome to the honorable speakers, Dr. Raphael Perez from CIRAD, France, and Dr. Mochammad Khairil from Universiti Putra, Malaysia, and my colleagues, Mr. Mohammad Roviq, Magister Science from Departement of Agronomy, Brawijaya University. And this webinar is an important forum and the first international online meeting that a part of AMYCOS. While AMYCOS itself is the program of our department that has become one of the training program for the lecturers, especially to enhance the capability of junior lecturers to be ready in the presenting their research in front of the international community. It is a day to discuss some topics with the foreign scientists. It is also a day when the student and the lecturer could ask questions to speakers that related the topic. And the last but not the least, I expect that the participating that join in this Zoom meeting could take the advantage of the knowledge sharing from the event. Finally, I wish you all a very successful webinar. Have a nice webinar. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Nurrahmi Abiarini, for the opening speech. The next one is our first session from Mr. Muhammad Roviq, Master in Plant Science. We also invite Mrs. Siska Fajriani, Master in Science, as the moderator. Well, our moderator today, Siska Fajriani, Master in Plant Science, is lecturer in Department of Agronomy, Faculty of Agriculture, Universitas Brawijaya, Indonesia. Her educational background are Master in Plant Science at Universitas Brawijaya and also Bachelor in Horticulture, Universitas Brawijaya. Her interest in microclimate and horticulture. And if you want to contact her, please contact her in siska.fp@ui.ec.id. Now, to Mrs. Siska, time and place are yours.
Okay, thank you, Ms. Paramita. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to today's let me introduce myself. My name is Siska Fajriani as the moderator in the first session. And our first keynote speaker is Mr. Mohammad Roviq, Master of Agriculture from Universitas Brawijaya, Indonesia. In this time, Mr. Roviq will deliver to us about growth differentiation balance of red peat to different source and supply of nitrogen. And this presentation will be delivered in 55 minutes and followed by 25 minutes for question and answer session. Before Mr. Roviq start his presentation, I would like to inform you about his curriculum vitae. Currently, Mr. Roviq is staff in the Department of Agronomy and also as an expert staff at the Faculty of Agriculture. His last education was a Master of Plant Science and currently undergoing a doctoral program in the field of agricultural science. And his research interest is plant nutrient and plant growth analysis. So if you will discuss with Mr. Roviq about the research interest, you will send an email to mohammadroviq@ub.ac.id. Without any further ado, I would like to invite Mr. Mohammad Roviq. Mr. Roviq, time is yours.
Thank you, Bu Siska, and good afternoon, Bapak Ibu, and all of students here. And we know that so many students in this and join in this meeting. I don't know what you want or what you need, but hopefully my presentation will give you any good information, or maybe especially for all of you that will start to conduct the research. Right, I will share a material that will I present today. Siska, do you can see my material?
Yes.
Okay. The topic or the title is a growth differentiation balance of red peat to different source and supply of nitrogen. Actually, this research conducted by four researchers. Beside me, and Ibu Prof. Elis Nihayati, Dr. Sitawati, and Prof. Marno, and also Prof. Sitompul. And this is about the actually how to choose some of that can increase the yield and also to prevent decrease of metabolite secondary. The outline of my presentation is consists of introduction, material and method, result and discussion, and conclusion and closing. So the introduction of my research was the report I've been so maybe about the 37 or 50 in Bangladesh, but in progressive countries is about 60 to it leads to phenomenon growth differentiation balance as a balance of regulation of Okay. Okay. So there is the objective of my research. First, to study the effect of different source and supply of nitrogen on growth, yield, and phytocyanin or
Committee will can help. Sorry. There is any trouble here, we will fix the problem in a minute. Sorry for a little trouble. We Mr. Roviq to join again. Sometimes the signal is not good.
Thank you, Siska. I'm sorry because I get some problem in my tools.
Okay, Mr. Roviq, please.
So this there are many objectives from this research. The first, to study the effect of different source and supply of nitrogen on growth, yield, and phytocyanin of red beet. And the second, to find out the correlation between the variables of growth, yield, and phytocyanin of red beet. And the third, to study the balance among yield, phytocyanin, and total phytocyanin. And the fourth, to study nitrogen agronomy efficiency on yield. And the last, to study the appropriate harvest time to increase the gain in yield, phytocyanin, and total phytocyanin. Material and method. Mas itu. Okay. The study was conducted at the experiment station in the University of Brawijaya in greenhouse area. in Malang area. And mantul suaranya. Saya dengar suara saya sendiri. Okay. Uniform and healthy 10 days old seedling were transplanted into plastic pot, and in each pot, plastic bag filled with topsoil and the soil mixed with compost and added with husk, with a was applied with treatment on 70 days after transplanted. The experiment was arranged according to randomized block design with pre-replication. The treatment consists of urea, ZA, calcinate, NPK, nitrat, and also NPK. And the second factor is the supply or dose. The first is 75 kilogram nitrogen per hectare. The second is 150 kilogram nitrogen per hectare. And the third is 220 kilogram nitrogen per hectare. And we give one treatment as a control. Observation about the plant growth consists of leaf number, leaves area, total plant fresh weight, and total plant dry weight, and observed with destructive samples on 28, 35, 42, 49, and 57 days after transplanting. And about the yield, consists of 3 variables. There are root diameter, root fresh weight, root dry weight, and different with the plant growth. We add the 1 days or 1 observer's time in 66 days after transplanting. And also quality, it means secondary metabolite, we observe in the same time with the yield, consists of beta-cyanine content and total beta-cyanine. Next, the data analysis. For the aims first is analysis by variance analysis data or ANOVA and smallest significant difference with the P 0.05 to compare the mean values between treatment. And the aim second, we analyze by correlation inversion two-way. And the third aim, the empirical model with inflection point and minimum or maximum Y in polynomial polynomial model. And the fourth, we analyze by mechanistic model with inflection time in logistic and beta model. And the fifth, with the mechanistic and empirical model, especially by logistic and polynomial in order to order. So the first problem, is there any and how is the response of growth, yield, and beta-cyanine of red beet to different source and supply of nitrogen? So this one is the result. We know that the application of nitrogen fertilizer at 150 kilograms and per hectare was able to increase leaf area higher than especially control and also in 75 kilogram per hectare, but not different with the 220 kilogram per hectare. And also differences in nitrogen source didn't cause a significant different effect or leaf number and leaf area. Is still in growth about the total plant dry weight and total plant fresh weight at 70 and 56 days after transplanting. We note that the plant fresh weight and plant dry weight in calcinate and urea application tend to be higher than other fertilizer. And also supply and fertilizer in 150 kilograms per hectare produce dry weight and fresh weight of plant, which tend to be higher than other dose. And about the yield of beetroot consists of beetroot diameter, beetroot fresh weight, and beetroot dry weight. We know that is on the graph, the result of fresh weight and dry weight of beetroot showed that the use of calcinate and urea was able to increase both higher than another fertilizer. And also about the diameter didn't show a different response regarding there was a bias maybe caused by a shape that was not actually symmetrical or full round in beetroot. So this is why diameter of beetroot maybe is not respond in significantly different. And plus the dose of 150 and 220 kilograms and per hectare resulted in higher fresh weight than those of lower end doses. And the last is about the quality. It means secondary metabolites, especially beta-cyanin content and beta-cyanil total. The result, the beta-cyanin content responses showed a decrease by increasing of dose. Yeah, but it's not significant. It is very possible that it was related to high coefficient of variance. Because maybe the high of variance in 21%, and the total beta-cyanin in application of calcinate and urea tend to be higher and only significantly higher than ZA and control. So the summary of first method in first problem based on the result of this condition study, it means in pot and in greenhouse in spacing 28 per 20 centimeter per segi. Calcinate and urea was one option from several nitrogen, which could increase yield and total beta-cyanin in red pit higher than NPK, NPK nitrate, and ZA. So we go to the next problem. The second problem is there the correlation among the variable of growth, yield, and beta-cyanin of red beets. This one is the data analysis by person, and the result is leaf area was a growth variable that was positively correlated to yield, but it didn't show a close correlation with the quality, especially beta-cyanin content. Fresh weight and dry weight of plant were not surprisingly when positively correlated with the yield and beta-cyanin total. And also beetroot fresh weight and beetroot dry weight which positively correlated with the beta-cyanin total. And BC, or beta-cyanin content, was the one and only variable that has a negative relationship with the other variables of growth and yield, but was not significantly statistically significantly correlated. Yeah. And BC was only positively correlated with BT. This means that the decrease in beta-cyanin content has the potential to decrease beta-cyanin total. This is why we must consider this fact. So the second summary, there are two conclusions. First, the increase in growth was positively correlated with an increase in yield, primary metabolites, but not significantly correlated with the decrease in beta-cyanin content as secondary metabolite. And beta-cyanin content was positively correlated with total beta-cyanin, so that was necessary to pay attention the limit where the increase in primary metabolite didn't decrease secondary metabolite significantly. So we go to the next problem. The third problem, is there a balance? And where is the meeting point for the balance between yield beta-cyanin and total beta-cyanin? Okay. We analyze by polynomial model or empirical model in order third or order 3. The result is based on the result on the first and the second analyze. The third analyze focus on calcinate and urea. So we don't want to discuss NPK, NPK nitrate, and also ZR because based on the ANOVA and LSD, the yield is lower than those in calcinate and urea. And based on polynomial order 3 modeling for yield and beta-cyanin balance, so that the estimated for the optimum dose of urea is was between 170 and 186 kilogram nitrogen per hectare, and the optimum dose of calcinate is between 166 and 210 kilogram per hectare. At the dose, the addition of beetroot fresh weight was able to compensate for the decrease on beta-cyanin level, as indicated by the high total beta-cyanin. So this one is the summary for that objective. That is based on the polynomial modeling, it was estimated that the optimal dose to produce high yield and total beta-cyanin were from 170 to 185 N per hectare for urea, and from 166 to 210 kilogram per hectare for calcinate. So the next problem is, what is the amount of nitrogen supply that achieve high agronomy efficiency? Based on the logistic and beta model, we note that the level of agronomy efficiency and the tree dose increase according to the time of growth at different rates. So we know that in 66 days after transplanting, the agronomic efficiency is higher than the 28 days after transplanting. This means it's like that. And the level of agronomic efficiency at the dose of 75 kilogram nitrogen per hectare of urea and calcinate fertilizer was higher than those at the higher dose. Although the beetroot fresh weight at the 150 and 220 kilogram per hectare was higher than those of 75 kilogram nitrogen per hectare. It means that the addition of fertilizer dose up to 2 or 3 times from 75 kilogram per hectare was not equivalent to increase the beetroot fresh white as high as the increase of dose. So this one is the summary for fourth objective. The level of agronomy efficiency of those 75 kilogram per hectare of urea and calcinic fertilizer was higher than those at the higher dose. So this one is the last problem. When is the appropriate time for harvesting due to rapid biennial crop? And is there any change in beta-cyanin content over time? So, as we know previously that the productivity of red wheat cultivated in Batu City, especially in high area, about 37 to 51 ton per hectare, and a productivity of red wheat in main producing country is 10 to 20 higher ton, 20 ton higher than in Batu City. But based on the simulation of the time of in harvesting time, we know that if we harvest in 49 days after transplanting, we only get the 1/3 ton from the 66 days after transplanting. Okay, why we can harvest in the 49 days? Because the diameter of red wheat, the market range from 5 to 10 centimeter, which in study occur at the age of 49 to 66 days after transplanting. And also yield potential at harvesting in 66 days after transplanting was 2 times compared to 49 days after transplanting. So it means that harvesting time determines how much yield was achieved. Okay. And the second simulation, we use the model. Consists of 3 models here. First, 2 models, polynomial order 2, and also use the logistic model. In general, beta-cyanin content tended to decrease in according to increase in plant age. Yeah. In black line, the total beta-cyanin value at 66 days after transplanting was very wide. It's, we can see about the quartile 1, quartile 2, and also maximum, minimum, and also mean and median. And based on this model, there were estimated that at 63 days after transplanting, although the total beta-cyanin at the high level, the beta-cyanin content has reached the lowest point. So harvesting at the time was considered quite good to prevent further decline. And the next simulation it is about the implementation of this research to farmer actually. By estimated in yield, we know that if we harvest in 49 day after planting, for calcinids, maybe we get loss, but for urea, we get gain, yeah, because the urea fertilizer was more profitable than calcinids. And we know that calcinid with 225 kilogram per hectare had the potential to suffer losses, especially if the yield was below 60 ton per hectare or harvest before 60 days after transplanting. The conclusion, calcinid and urea is one option from several nitrogen which can increase yield and total beta cyanide in red pit higher than NPK, NPK nitrat, and ZA. And the second, the increase of growth was positively correlated with the increase in yield or primary metabolite, but not significantly correlated with decrease in beta cyanide content or secondary or as secondary metabolite. Based on the polynomial model, it is estimated that the optimal dose to produce high yield and total beta cyanide are between 170 and 185 kilogram N per hectare for urea, and 166 and 210 kilogram nitrogen per hectare for calcinid. The level of agronomy efficiency at the dose of 75 kilogram nitrogen per hectare of urea and calcinid fertilizer was higher than those at the higher dose. Beta cyanide content tend to decrease in according to increase in plant age, and to prevent the total beta cyanide from decreasing was harvest at the around age of 63 days after transplanting. The use urea fertilizer was more profitable than calcinid. That's all, and special thanks for Prof. Sitompul for directing the research substance in peace. Thank you. That's all of my presentation today, and back to Bu Siska.
Okay, thank you, Mr. Roviq, for the nice presentation. Now we are coming to the question and answer session. I think the audience will ask a lot of questions for Mr. Roviq because the topic that you deliver is very interesting. I will invite 3 questions from audience in this session. The audience will raise hand to deliver the question, or can write the question in the chat box and I will read the question. Maybe somebody want to ask? Okay, Hana Shiva.
All right, thank you.
You can unmute. Okay.
So, yes, my name is Hana Shiva. I'm from student of Department Agronomy. So from the research by Mr. Roviq delivered before that, said that calcinid and urea is an option from several nitrogen which can increase yield and total beta cyanide and red pit higher than NPK, nitrat, and ZA. But at the fifth problem, it will conclude that the use of urea fertilizer was more profitable than calcinid. So my question is, if we want to use another nitrogen supply instead of urea, is there anything we should pay attention to stay profitable? If we don't, urea is more profitable. How if we want to use another nitrogen supply? Maybe it's my question. Thank you very much.
Okay, thank you, Hana Shiva. Okay, Mr. Roviq, you can answer the question from Hanasifa, please.
Because
Uh,
I don't hear anything in my tools.
Okay, Ms. Hanasifa can help the
All right. So
ma'am, sorry, repeat the question.
I'll repeat the question. So from the research by Mr. Roviq delivered before that, calcium and urea is one option from several nitrogen which can increase yield and total production in rapid. But the last, urea is use urea fertilizer is always more profitable than a calcinate. So my question is, if we want use another nitrogen supply, is there anything that we should pay attention to make the rapid is stay profitable? Maybe that's my question. Thank you.
Yeah, good question. Thank you, Ms. Hannah. Yeah, there are many considerations actually if we want to choose what the fertilizer is. And the first, in the first time, I'm sure that technically urea and calcinate give the good or the best response for some of a variable, especially in yield and also in beta-cyanine total. But in about another thing, we know that the price of calcinate is about 44,000 per kilo, but if we use the urea, we only pay about 11 or maybe 11 or amount 10 10,000 rupiah. And also the using calcinate because they only contain about 27% of nitrogen, and compared with the urea that nitrogen content is about 46. Yeah, 46. We can use urea less than a dose of urea less than the calcinate if we need the nitrogen in the some dose. That is, Hannah.
All right, thank you for the answer, Mr. Roviq.
You're welcome.
Thank you, Mr. Hannah, for the question. And we're going to the next question. Maybe Professor Arifin will give the question for Mr. Roviq. The audience. Okay, I want to ask you, Mr. Roviq, as we know that rabbit are generally grown in the highland, yeah. Is your research also conducted in the highland or the lowland?
Yeah. Okay. Thank you to Siska. My research conduct at the lowland, especially about 400 above sea level. In what is it? What is the name? the Griasanta garden, Siska.
And then the result in this research could to be applied in the lowland or in the highland or the two place.
Yeah, maybe it's depend. First, if we want to business about the beetroot, actually, we know that the highland maybe is appropriate place to get the higher yield. But if we see in the other side about the relation of yield and also the metabolite, secondary metabolite, we know that we must pay attention about the increase of yield that maybe can decrease the beta-cyanin content. So that's why we must make a point of thinking about the primary and secondary metabolite. This means, or this one is the important thing when we want to cultivate the red beet, Luciska.
Thank you, Mr. Rafiq. And I will ask you about the reason you choose the calcinate as a source of nitrogen for nutrition for red beet. What the reason you choose calcinate? Yes.
Okay. Yeah. Yeah, we know that so many fertilizer or so many source of nitrogen in the market actually, and sometimes some of people or some of farmer say that this one fertilizer can increase the yield and also the beta-cyanin content, etc., etc. So it is why we must test some of the some of the fertilizer, yeah, and as we know that the yield and also beta-cyanin total from calcinate as high as urea, but in the other side we must pay attention about the price of calcinate. That is to Siska.
Okay. Okay, thank you, Mr. Rafiq, for the nice answer. I will invite the audience to ask Mr. Rafiq. You will write the question in the chat box or raise hand. Okay. The question is from Fatul Rahman. I apologize in advance. My name is Fatul Rahman from the Faculty of Agriculture, Universitas Brawijaya. May I ask, sir, can the result of this research be applied to various land conditions, such as land that has excess water or lack of water? Mr. Rafiq, please.
Yeah, actually, we should pay attention in the stress condition, like maybe land condition, especially in dry land or excess water and something like that. And of course, this research especially about the metabolic secondary, will affected by the under stress. So this result of this research cannot apply directly in under condition, especially in a stress condition. That is, Siska.
Maybe any feedback from Fatur Rahman? You can unmute your microphone and give a feedback for Mr. Roviq. Fatur Rahman. Okay, we go to the next question from Angelique Novalia Tambun. In the presentation, it was explained that beta-cyanine content is positively correlated with total beta-cyanine. So it is necessary to pay attention to the limit where an increase in primary metabolites does not significantly reduce secondary metabolites. So my question is, what restrictions should be made so that secondary metabolites do not decrease when primary metabolites increase? So you can answer the question.
Yeah, actually, you answer your question by yourself, right? Yeah, we know that we must pay attention if you try to increase the primary metabolic, especially about the yield, or especially about the beetroot fresh white, because in the same time we know that the beta-cyanine content, although there is not significantly correlated with the increase of yield, but have a negative correlation, yeah, but not significant. It is show that the metabolic secondary has negative relation with the yield, and this is why we try to give some supply of nitrogen in 75, 150, and also 225. And we note that above 125, the yield is not significantly increased more. Yeah, or maybe it is optimum level is about the in ring in between 150 and 275. It is about the yield, but we know that the decrease of beta-cyanine is compensated with the increase of the yield. So the total beta-cyanine is still in higher value or in higher number of beta-cyanine content or beta-cyanine total. This one is the meeting point of beta-cyanine content and yield.
Okay, for Angelique Novalia Tambun, you give the feedback for Mr. Roviq. Or maybe you have any questions. Okay, continue for another question from Intan Nur Aini. Permission to ask why doses of 150 and 225 kilogram nitrogen per hectare resulted in primary metabolic yields. Whether it be correlated when we increase the dose, the resulting BRFW will also be higher. Thank you.
Yeah, the point is the yield will equivalent with the dose, but after the the point, it means the maximum point, the yield will not increase again or increase more. Yeah, it is like the maximum capacity of zinc. It means the beetroot fresh white have a maximum capacity to fulfill by biomass. The first answer is like that, and the second opportunity or the second option is with the spacing in 20 and 20 centimeter per segi. We know that maybe some of variable like what is it like leaf area of leaf number, especially leaf area, I think will what is it will competitive will get a competitive point, right? And some of leaf will close or maybe will what is it will get less of sunlight. So this means that the rate of source or photosynthetic process or photosynthetic will not increase again, because so many conditions that not support for plant to grow up and also to get some of environment factor in optimum level. So it means that the increase of fertilizer for some plant in general not always followed by increase in yield, because some conditions like the competition, and also this one is must be give our attention beside about the maximum capacity of zinc. This one is my that are my answer.
Okay, thank you, Mr. Rafiq. Okay, thank you for the answer, sir. Tanah I will give a feedback. Okay, audience, any audience can ask the Mr. Rafi. Okay, the question from Uswatun Hasana. I will read the question. How do we produce as much beta cyanide as urea and calcinate do, but using NPK, and is it impossible? Should we increase the dose? Will it be as optimal as urea and calcinate? Mr. Rafiq, you can answer the question from Iswatun Hasana.
Thank you for the question. Yeah, based on the result in the same dose of several fertilizer or N sources, we know that the productivity or yield of NPK and NPK nitrate is lower than those of or in calcinate and urea, right? And of course, the addition number of or addition dose of NPK will increase the yield, but still in compare with the NPK. But when we compare with the yield of calcinate and urea treatment, we know that the yield of NPK is still lower than yield in calcinate and urea. And after the dose, the maximum dose, it means 225 kilogram per hectare. We can see that the yield is not increase more, right? Both in calcinate and also in urea, and also in NPK or another fertilizer. And we know that the supply of this fertilizer is very high in the space on the pot, in only space in 20 20 and 20, right? So if we want to get the profitable yield, maybe it is appropriate for us to try in lower dose, something like that, rather than we try in higher dose than 225 kilograms nitrogen per hectare.
Okay, thank you, Mr. Rafiq. Maybe Iswati Praksana want to give feedback. Okay, we go to the next question from Mahavira. Good afternoon, sir. Sorry, I can't turn on the mic because my internet connection is not good. The question is, in the graph, it is stated that the beetroot fresh weight and beetroot dry weight indicate that calcinate and urea are able to increase higher than other fertilizers. What causes this to happen?
What causes?
Yes.
Yeah. Maybe the question. Can I simplify? Why? Yeah, because we know that plant in general need a minimum level of fertilizer as the, what is it, the important factor to grow, and after grow they will transport the photosynthesis in organ that accumulate the photosynthesis like beetroot. So it means that if we only give the lower factor, especially fertilizer, below the minimum level of plant need, it means that the growth of plant will not maximum or optimum, right? And because of that, or due to this condition, the yield will not be high. So it means that, yeah, actually we need the point or the dose, or how to know how to get the appropriate dose to get the higher yield. Yeah, and because of that, we do in 3 level, 75, 150, and also 220, and we use control with the zero addition of fertilizer. And after some of analysis, we know that the point of minimum, or maybe the optimum dose, is about the 115. Below or above this dose, we need not efficient in yield, and also maybe not efficient in cost, something like that.
Okay, thank you, Mr. Rafiq. We have 3 questions again from Jamilatun, Ragil Gibran, and from Andini Mustikasari. I will read the answer from Jamilatun Munira. The question is, may I ask her, is there any methods to make the red beet harvested more than once in 100 days after planting without decreasing the quality and quantity of crop? I can repeat the question. Maybe Mr. Rafiq.
Yeah. The question is, harvesting is more than, yeah, twice harvesting?
Yes. Yes.
Oh, yeah. The characteristic of the beetroot is when plant only have one taproot, yeah, taproot or beetroot, yeah. If after we harvest, or we put harvest the beetroot, this plant will not produce another taproot, right? And if you try to characterize this plant, it is more beneficial for us to plant another seed rather than try to like grafting on something like that. So as we know, today we cannot harvest this plant twice or more than one harvest time.
Okay, thank you, Mr. Roviq. The next question is from Andini Mustikasari. You want to explain that that in the simulation, the harvest time determines how much competition is achieved. My question is, what are the competition points in the harvest time simulation? Can you explain the simulation maybe?
Okay, my question is what different point. Okay, it's on the model, especially logistic and versus logistic model for yield growing up, and also by the model the beta cyanine content that the decrease by the time. We know that the time to harvest is 63 days after transplanting, but it depends, right? It depends in what where the place that you can cultivate this plant, and also what your method by sowing the seed directly to the land, or by what is it, like transplanting of seedling, yeah, because after you transplant the plant get the stress as adaptive condition before they grow normally and healthy, right? So it's mean based on or in my research, the point, the meeting point of the beta cyanine content that tended to decrease by the time, and also the yield increasing by the time. So the meeting point is in 63 days after transplanting. But maybe if you conduct another research, you will get another time or another what is it, period to get the meeting point. That is my answer.
Okay, thank you, Mr. Roviq. Lots of questions for you because the topic is very interesting. Yeah. Sorry, I misreading the question for Ragil Gibran. I will read the question. In the third point of conclusion, there's a dose on how much urea and calcinate we can use to produce high yields and total beta cyanine. I noticed there's a huge range in those doses. So my question is, what caused that huge range in those doses? And what happens if we use too much calcinate?
Okay, based on the data, actually, we have the ring of dose. The first one is about the yield, actually. Then the next dose, it means like this, 100 and 170. up to 186, it means that the first first enter of dose is in the lower dose, and the what I said, in the highest dose is the last mentioned like this. Okay. The first dose is about the yield actually, and the last dose is about the what I said, like what is it? Actually, this one is beta-cyanine content, right? It's not about the beta-cyanine total. It's not about the beta-cyanine content. It's been that the tolerate point, right? Because when we fertilize or apply some nutrient to plant, so many factors that can cause the bias. For example, the characterize of media, and also maybe about the size of plant and age of plant, something like that. So it means that this point is the range of dose that can be tolerated by plant, or maybe optimum by plant, both of yield and beta-cyanine total. It means that this one is the ring in between 150 and 225. So actually, although we only have the treatment only give a treatment with the 3 doses. It means 75 and 150 and also 225. Actually, we cannot say that this one is the best dose actually, because if we try to analyze by the model, we get a graph, and the graph show that the relationship between X factor and Y factor and or independent factor and dependent factor is not always constant like linear regression. That is like the non-linear regression, especially about the dose is polynomial order 3. So if we use polynomial order 3, we can get some point. First is point of inflection, that is like what is it? The point for minimum rate or to maximum rate or reverse, something like that. And also we can get the maximum point or minimum point. So if we use the polynomial order 2, but if we use polynomial order 3, yeah, but if we use polynomial order 2, we only get the 1 point. It means the maximum or minimum point. It means that the level of fertilizer that apply in our research actually is not the what is it fixed point to achieve the higher or optimal level, right? So we should analyze by another approach, like the model, to get the point. And this in this my research or our research actually, we get that the optimum level is about the ring. It's not only one point, because we have two variables that what is it that make beta-cyanin total. It is beta-cyanin content and also the yield. That is my answer.
Oh, it's a long answer, but it's good information for us. Yeah. And the last question in this session, I want to read the question from Abdurrahman. My name is Abdurrahman. I want to ask in conclusion, it is explained that the beta-cyanin content tends to decrease according to the age of the plant. And to prevent the decrease in total beta-cyanin is to harvest at around 63 days after planting. Is there anything else that causes harvesting to be done at the age of 63 days apart from preventing the decrease in beta-cyanin content?
Yeah, it is a question that related with our research actually. Yeah, first we need a higher quality of beta-cyanin, right? But another side, we must what is it? Consider some point, especially the yield compared with the cost. It means the yield is revenue, yeah. And we know that if we use calcinate, we must harvest in more than 60 tons per hectare, because the using of calcinate is more expensive than urea. And also it's about the time of harvest. We can get the 60 ton per hectare in yield is more than 60 days after transplanting. So that is the some point if we want to harvest based on maybe the price in cultivation and also the quality of target, yeah, target of quality, yeah, right? So the 63 is suggesting day after transplanting that you want to we want to harvest red pit in my experiment, right? If you want to if you conduct another experiment in another condition, sometime in different environment or in stressful condition, or the different level above sea, maybe something like that, maybe the edge or the meeting point of harvest will be different with our research in my experiment, right? That is to Siska.
Okay, thank you, Mr. Rafiq. Turns out there is still one more question for Mr. Rafiq. I'm sorry, this is the last question. Please, Mr. Rafiq can give the short answer for this question. In the last conclusion, stated that the use of urea fertilizer is more profitable than calcinate. So my question is, what are the points that make urea fertilizer more profitable than calcinate?
Yeah, based on our simulation, we entry some point, especially the price of calcinate. Yeah, calcinate compared with urea is more expensive. Yeah, it is. We know that the price of calcinate is about 44,000 rupiah, but the urea is only 11,000 rupiah, and also urea is more. The dose of urea is less than calcinate in the some nitrogen yeah. So because the nitrogen content of urea is higher than calcinate, nitrogen contain about 46% of urea contains 46% of nitrogen, but calcinate only 27% of nitrogen. That's why the dose of calcinate is higher than urea in the some level of nitrogen need, but and in the other side, the price of calcinate and also higher than urea. This is why the urea is more profitable than calcinate if we use if we compare both of them. That is, Siska.
Okay, thank you for Mr. Roviq. I think it's enough for this question and answer session. Okay, that's all for first session. Thank you for Mr. Roviq for delivering the interesting topic in this afternoon, and also thank you for all audience. Hopefully it will be useful knowledge and information for us how to choose the different source and supply of nitrogen in the growth of red beet. And as a tribute from us, here we present a certificate to Mr. Roviq as a keynote speaker in the Agronomy Conference Series, International Webinar from Department of Agronomy, Faculty of Agriculture, Universitas Brawijaya.
Thank you, Siska.
Okay. This is end of the first session, and I will hand over to the MC, Mrs. Permita, please. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Well, that's very interactive session. Thank you, Mrs. Siska, for moderating the first session, and Mr. Mochammad Roviq for the interesting topic. Now, I will invite Mr. Roviq and Mrs. Siska to take a picture together, please. For take a picture together, Mr. Roviq and Mrs. Siska. Okay, get ready. 1, 2, 3. Okay, already. Thank you so much. Well, ladies and gentlemen, now we come into the second session. Mr. Khairil Mahmud, Ph.D., as the speaker, is already with us, and Dr. Adistiawan will guide this session. Okay, Dr. Adistiawan is our moderator in second session, is lecturer in Department of Agronomy, Faculty of Agriculture in Universitas Brawijaya, Indonesia. His educational background are Ph.D. Environment and Nutrition Science at University of Miyazaki, Japan, and Master in Plant Science at Universitas Brawijaya and Bachelor in Agronomy at Universitas Brawijaya. His research interests in tropical environmental science, sustainable agriculture, agroecology, agroforestry, and also forest ecology. If you want to communicate with him, please contact in adistiawan@ub.ac.id. Now to Dr. Adistiawan, time and place are welcome.
Okay, thank you very much, Bu Paramita or Mrs. Paramita. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to the Agronomy Conference Series, or we call it AMYCOS. And I would like to introduce myself. I am Adistiawan, and I will be a moderator today. You will be here a presentation from the Honorable Dr. Khairil bin Mahmud on this very timely subject. And finally, after we are correspondent via email, I meet together at this event with Dr. Khairil. And however, before we get started, I want to take a few minutes of your time to introduce our speaker today. Please to show, okay. The Honorable Dr. Khairil bin Mahmud is a speaker in this session. He is a Department of Crop Science, Faculty of Agriculture, Universiti Putra, Malaysia. And his research interest is botany or agroecology, and you can contact with him by email. The email is Khairil Mahmud, UP2, and or qualification obtained. He got the doctoral or Ph.D. course by School of Biological Science, University of Aberdeen, Scotland, Kingdom, UK in 2018. With specialization is ecology, and he got from master from Faculty of Science and Technology, Universiti Kebangsaan Malaysia in 2010 with specialization botany and Science and Technology, Universiti Kebangsaan Malaysia in 2007. Specialization as biology. I get a publication list from him, and the publication and mostly about the phytoremediation.
Okay, sorry for this inconvenience. There is trouble in our signal, so please wait. We will fix it very quickly.
Yeah, sorry, our connectivity is not good, so we will repeat it once again. For our publication list, I get Dr. remediation topic. So I think it's very interesting if we hear about this topic today. I think Indonesians and another country like Indonesia, P2 remediation topic, it's essential for cleaning our environment. So, and I think it's very important today to hear about this topic. Dr. Khairil, we have a two part in this section. The first section is presentation from you, around 45 minutes. And the second one is a discussion, approximately 25 minutes. And the last section is certificate submission. Okay, now please welcome the Honorable Dr. Khairil, who will be speaking to us on P2 remediation, how plant help in cleaning up the environment. To Dr. Khairil, please.
Okay, Assalamualaikum and good afternoon to all of you. And I would like to thank to University of Brawijaya, to the HOD, okay, Head of Department, Dr. Nurrahmi, and Dr. Damaduri, as the Dean of Brawijaya University, and of course to my friend Dr. Adi Setiawan. So thank you so much for inviting me and giving me a good opportunity to give a talk about the fight to remediation. Let me share my slides again. So I would want to introduce myself because, yeah, thanks to Dr. Adi Setiawan has introduced myself just now. So this is my topic for today, how plants help in cleaning up the environment. So when we talk about phytoremediation, what is phytoremediation? And why nowadays people are concerned about our environment, about our crops, you know, crop safety. Why is it? Why it is important? Which plants we use for phytoremediation? How it works? How about the mechanism? Okay, and which elements that we use the plants to take out the heavy metals? And is it a green technology? Okay, so let's start with this. I mean, high levels of heavy metals and metalloid in soils have become a significant problem globally. Okay, including Southeast Asian region, Indonesia, Malaysia, you know, Thailand, Laos, Cambodia, and yeah, heavy metals and metalloids are found in the soils through natural process and human-made activities. So these are some of the information, okay, some of the source of contamination where they came from the natural sources and also from the anthropogenic sources. So the anthropogenic sources is the heavy metals, okay, they came from the agricultural sources, okay, inorganic and organic fertilizers, okay, liming, animal manure, okay, pesticides, herbicides, and fungicides. So it also came from the industrial sources, mining, okay, transport of course, smelting of metals, okay, recycling of metals, domestic effluents, okay, household pipes used for supply of water. It contains zinc, okay, copper and chromium, sewage sludge, zinc, lead, and chromium, and of course, waste generated by household activities. Okay, so these are some of the information about the source of the contamination. And yeah, the term heavy metals actually, it refers to the metallic chemical element that has a relatively high density and is toxic or poisonous even at low concentrations. Examples of heavy metals include mercury, cadmium, chromium, thallium, okay, lead, and also one metalloid which is toxic, okay, to not to humans but also to many organisms, which is arsenic. So, and a metalloid is an element that has properties that are intermediate. So they are intermediate between those of metal and non-metals. So metalloids can also be called as semi-metals. So this is some example of the arsenic contamination worldwide. Okay, this is happened in groundwater in the world. So where we are, okay, Malaysia, Indonesia, okay, we have some kind of the information, some kinds of the contamination about arsenic, especially in the South Asia countries like in India, Bangladesh, and yeah, we cannot ignore this. This is very important. Why I say this is very important? We are trying to get our better food, a safer food, okay, for the food safety. We must try to have a better product or crops which free from this kind of contamination. And heavy metals contamination in the environment, they came from, like I said, they came from the industrial, okay, from the mining, from the agricultural sources and all this, okay, they came and enter the water, and what happens? Sediment and so on. So this is some of the pictures, okay, that just to explain what is actually happened, okay, how about the heavy metal contamination in the environment. This is what I said, it came from many sources, okay, the lead, okay, where does it came from? Automobile exhaust, okay, chimney, gasoline, cadmium, where we can get this from the earth crust, okay, metal coating, batteries, okay, batteries production, okay, copper, jewelry industry, fungicides, and all this. This is actually happen nowadays in our environment. Yeah. So these metal contaminations in the soils are generally toxic to the crops, and this may later unintended health. Okay, it gives a bad effect to the health, to the human. Okay, upon the ingesting the crops contaminated with heavy metals. This is what happened actually. Okay, in some other countries in Asian. Okay, in Asian countries. Okay, so this is how the plants, okay, they absorb the arsenic, okay, absorb the heavy metals, and what happened, it may transfer into our food chain. Okay, and so the traditional remediation technologies such as soil washing, capping, leaching, soil flushing are generally less efficient, costly, causing soil destruction and produce secondary pollutants. So therefore, we come out with an idea of phytoremediation, and it has been done in such many countries before. Okay, phytoremediation has become an initiative, effective and affordable technology used to remove all these contaminated from all these pollutants, okay, from the contaminated soil. So it is considered as the environmental friendly and cost-effective method, okay, where we use the plants, okay, to clean up the environment from the soil, from okay, clean up the contamination in the soil, sediments, and water. So plants we use for phytoremediation, including grasses, shrubs, trees, right? Okay, which are associated with the microbes, okay, through the degradation, accumulation, and stabilization of the contaminated soil. So we can see the association between the plants and the microbes, how they work, okay, in order to clean up indirectly, okay, the pollutants in the contaminated soil. Then some plants, they have the ordinary abilities to accumulate high concentration of metals, okay, to accumulate the toxic metals, and they accumulate them without having injuries. And these are called as the hyperaccumulator. And hyperaccumulation trait in plants are now becoming of interest by many researchers for the advanced research in phytoremediation technology. I'm doing my research, me and my team doing our research on phytoremediation. And my PhD, I did my research on Melasthema malabatricum, where it is the aluminum hyperaccumulator. So these plants, they accumulate high concentration of aluminum. Okay, we know that aluminum is toxic to most plants. Okay, toxic to crops. Okay, but these plants can absorb, they can accumulate high concentration of aluminum without having any injuries. So this is the what we call it, the group, we call it as the hyperaccumulator. So let's discuss about what is metal hyperaccumulators. So hyperaccumulator term is clarified as a unique plant. They are unique. Okay, they accumulate particular metal or metalloids in their living tissue. Okay, they keep them in their tissues. Okay, to a certain level that may be hundreds or 1,000 times greater than non-hyperaccumulator or other plants. Okay, so let's say some plants, they are sensitive to these metal elements, but not to the metal hyperaccumulator plants. They can absorb it. They can tolerate to that kind, a certain kind of environment. Okay, the first term had been introduced by Brooks in 1977. It was few decades ago, okay, to describe those plants that take up and accumulate more than 1 milligram per gram metal dry mass in the leaves. So what is the association between hyperaccumulator and phytoremediation? So plants actively take large amounts of one or more heavy metals, okay, from the soil without showing a phytotoxicity effect. So they are actually useful for the phytoremediation. So that is trait. Okay, the hyperaccumulators, these are the traits that certain plants only, they have this kind of trait, but very useful in cleaning up the environment. So this is the threshold values. Okay, this is threshold values provided to define the hyperaccumulation of other heavy metals or metalloids based on its specific phytotoxicity set at more than 10 milligram per gram. Example for manganese, zinc, more than 1 milligram per gram. For aluminum, okay, not more than 0.1 milligram per gram, okay, and all this for cadmium. So this is actually the threshold, okay, from one of the research where very useful to identify at what level the plants can be considered as hyperaccumulator, okay. And one question when we talk about the hyperaccumulator, how they accumulate these metal elements. Okay, remember the accumulation of minerals, okay, and also the water is transported by using the xylem, by the xylem. So they came from the soil, the water been absorbed, and they are combined with the minerals and also the metal elements. So the xylem loading, the heavy metal will across the water impervious barrier Caspian strip, which helps metals ion to transport into the shoot by symplastic pathway. So, and then remember we have two types of pathway. One is apoplastic, the other one is symplastic. But whenever they are reaching the barrier of Caspian strip, they will pass it by the by this, what they call it, symplastic pathway before entering the xylem. So all these metal elements, the ions, they will come and entering the xylem through this symplastic pathway. Okay. On the other hand, root to shoot translocation, from the root to the shoot translocation of the large level of heavy metals in the xylem loading is enhanced by constitutive overexpression of gene coding. So gene also play important roles in accumulation of metal elements. For example, genes of HMA4 involve enhancing cadmium and zinc efflux from root symplast into the xylem vessel and promoting metals tolerance. So this is how certain God creates certain plants. They have this kind of they can adapt to this kind of metal elements and environment. So basically, when we talk about the mechanism of metal uptakes in metal accumulators, they contain 4 elements. Right? How they accumulate the metal elements from the soil, how the plants can use this for cleaning up the environment. The first from the accumulation in plants include metal bioavailability. The second one is root uptake. The third one is xylem loading. And the last one is detoxification or sequestration. So let's discuss about this. What is this? Extract and accumulation in this process involves several stages. Yes, of course. The extraction, again, cleaning up the process, they have several stages. Some heavy metals or metalloids, they present as soluble form. And in soluble form, that is unavailable for plant uptake. And this causes the plants to release variety of the root exudates, which can change the rhizosphere pH and increase metal solubility. And what happens next is the metals, which bioavailable to the plants, will then be absorbed into root cells, like I said, by using two types of apoplastic and symplastic pathway. So they are entering the roots through this two type of pathway. And the entering of metal ions inside the root cell surface by apoplast or symplast is dependent on the types of metal and the plant species. Not all plants, they can absorb metal elements. Not all plants can absorb aluminum. They are very specific. This trait only can occur at certain specific species. Okay, they are specialists. They are not the generalists. They are specialists in accumulating certain metal element. And detoxification and sequestration. So when the metal elements enter the xylem, okay, or we call it as a xylem loading. Detoxification and sequestration will follow. Okay, this is the process where the converting metal bioavailability into less toxic compounds. Why? Because the plants have the ability to change the toxic compounds and it becomes less toxic. And sometimes it's useful for the plants. In my study, I found that aluminum is toxic to most plants. It's toxic to crops. For your information, aluminum is the limitation factor for crops growth. Okay, but this aluminum accumulator, they can change the forms of aluminum and it becomes less toxic, and they keep them in the vacuum, they keep them in the tissues of leaves, okay, stem and roots, and they don't have any kinds of injury system or toxicity symptoms. Metal translocation from root to shoot is also mediated by organic acid, such as citrate, histidine, and molit. Why? It is to convert the toxic compounds to become less toxic, suitable, and harmless to the plant. That is how they change it. Again, they change the form to become less toxic. Sequestration is the final step, okay, of accumulation, in which the metal ions are away from the cellular process and occurs in plant vacuoles, or at least damaged parts such as epidermis, trichomes, and even cuticle. And it also can be the compartment, okay, they can be stored. The elements that can be stored in the leaf petioles, leaf sheaths, and trichomes, where heavy metals cause less damage to the plant. So that is how the movement, how the plants get the process of accumulating the metals from the root. It passed to the shoot. So certain plants, they are specific. Okay, they are specialized in taking up certain metal elements, okay, certain heavy metals. And basically, when we talk about phytoremediation, they must involve several parts, other parts of for phytoremediation, such as phytoextractions. This is the plant's roots, absorb contamination from soil and translocate it into the harvestable shoots of plants. So where does it came from? So we can see here, so the phytoextraction, they absorb from the roots until they keep them in the shoots. Okay. Phytostabilization. stabilizing soil contamination by complexation, precipitation, or root absorption rather than clean up. So they phytostabilization, they just keep them in the soil, but they make the soil become more stabilized. Okay, and rhizofiltration, okay, is removing pollutants from contamination sites by plant roots. Okay, so this happen in the plant roots. Okay, during rhizofiltration, the contamination absorbed either onto the root surface or absorbed by the roots, and phytovolatilization is accumulates the heavy metals and transforms the HM or heavy metals into a volatile form or less toxic substance, and subsequently released into the atmosphere. So this time it removed to the atmosphere by the transpiration process. Remember, the transpiration process is very related with the moving of water from the root to the shoot. That is how they transfer the heavy metals. But in primarily used in the removal of mercury, wherein mercury ion is transformed into the less toxic gases elemental mercury. And lastly, parts of the phytoremediation is the phytodegradation. This is what we call it, the plants and microbes in the soil to break down the pollutants or any other complex organic to less toxic substance via the metabolic process. So when you talk about phytoremediation, it covers all this. Okay, rhizofiltration, it can be phytovolatilization, okay, phytodegradation, and phytoextraction. And why phytoremediation? Why we want to do this? Okay, we are using the plants for cleaning up or detoxify the sites. Okay, so it is cheaper and it's more environmental friendly compared to other soil washing, okay, using other chemicals. At the end, the most expensive, the expensive one will become, we will have another issue. But using plants, okay, the cost is cheaper and they are more environmental friendly. For example, on abandoned mine sites, it is not uncommon for metal concentration in the surface soil to be one or more orders of magnitude higher than in normal soils. Yes, in such cases, many plants cannot grow on the sites that are left behind. Yes, I did some visit on some mining areas. Not all plants can survive. So the mining, they just dig the soil, and then the nutrients will different. Okay, some metal elements will occur. Okay, either in the high or low concentration. But I believe it was reported that some mining area, they have high concentration of certain heavy metals. Okay, some species, however, not only grow and thrive on such sites, but they take up the contaminants. So that's how the removing process takes part. Okay, removing the pollution from the soil and making the site more amenable to colonization by other plant species. That is why we need some kinds of what we call it, the phytoremediation process or phytoremediation activities. Okay, and currently more than 450 plant species from at least 45 families were known to have a potential for phytoremediation. We sometimes are not aware. God creates many plants which are useful for us. Okay, where until now some of the plants we cannot figure out the purposes. Okay, but actually in phytoremediation we have more than 450 plant species which were useful for phytoremediation in cleaning up the environment. So reducing the entry of toxic elements to the human food chain. Okay, this is some the statutory maximum annual metal loading rates over 10-year period of agriculture soil. Okay, this is some of the information useful, okay, in for soil according to UK, okay, USA, where we can find out whether it is critical or non-critical. Some natural soils can contain high concentration of mineral elements that are potentially toxic to plants and animals. For example, acid soils have excessive aluminum and manganese phyto availability. Serpentine soils can have excess nickel, cobalt, or chromium concentration, and seleniferous soil contains excessive selenium concentrations. And for your information, our soil in tropical region is acidic. When we talk about soil acidic soil, it contains high concentration of aluminium. Okay, and yeah, aluminium is one of the limiting factor for the crops. That is one. This is the topic of aluminium itself has brought many interest from the researchers, okay, worldwide. And reducing the entry of toxic elements to the human food chain. So animal manures, okay, slurries can contain significant quantities of cadmium, chromium, lead, again, cobalt, zinc, and so on. So similarly, sewage sludge can contain high concentrations, okay, of lead, okay, and all other heavy metal elements, and also the human pathogens. So composted municipal solid waste is frequently applied at high application rates, which can result in large amount of these metal elements. So when the industrial waste, okay, such as food waste, paper sludge, and fly ash can also contain significant amounts of potentially toxic elements, we sometimes we might not know that these elements, okay, sometimes they use it for the recycle, for the soil, for the crops, okay, for the plantation. So in many countries, legislation limits the quantities of heavy metals applied to soil on which edible crops are grown for human consumption. Why? Because they don't want to allow this happen, the transferring the heavy metals or toxic element metals to our food chain. Okay, there's a lot of issue coming in about the problem or issue related to the heavy metals, related to the food safety. It is important that these limits are followed to maintain both crop production and human health. So this is some example, okay. Arsenic in that chicken, okay. So they produce what they call it chicken, okay. They provide the food for this chicken, and but then it contains the arsenic in the chicken, and arsenic in drinking water poisoning 140 million people. Okay, this was reported in London. Okay, problem occurs mostly in Asia, and it could lead to higher rates of cancer. This is why we're concerned about the food safety. We might not know. We use the contaminated water, contaminated soils, and we start and we do the plantation, crop plantation, and you get the water supply, but we might not know that the soil or even the water is actually contaminated with the metal elements, heavy metals. And in long term, it may cause lots of issue, especially about the health, human health. And toxic pollution found in sea near waterfront site, okay? And closer to zero, FAO, FDA's action plan for reducing exposure to toxic elements from food for babies and young children. So this is about the food safety. It's a big issue, okay? We are facing with the food security, okay, with the global crisis, okay? People, okay, some countries, they stop sending exporting the wheat, okay, the what they call it, the oil palm, when while India and Pakistan have stopped exporting their goods, their what they call it, the wheat. So but actually we cannot ignore the food safety, because in the long term it may cause other problems if we not solve the issue like the contamination of pollutants in the soil and water. So this is one example, okay. These are particular concerns about arsenic concentration in paddy rice, especially in South Asia countries such as Bangladesh, India, and China. Okay, maybe we might not know what happened to our country, Indonesia, Malaysia. If we cannot solve this issue, we're not concerned about our environment. We must do something to avoid from this issue happens to our country. Again, the flooded paddy conditions led led to the mobilization of arsenic, which is taken up efficiently by rice roots through the silicon transport pathway. So rice, they absorb anything. So they absorb arsenic. They don't care. Okay. These monocot plants, they don't care. So they absorb it. And what happened? It entered the food chain. And this is the result if we do not, or they call it, we ignore the importance of environment, okay, the pollution, okay. We need to do something to avoid this from happening to our country. And feed regimes can be adopted that results in lower concentration of such elements in animal manure. Okay, when municipal compost are applied to agricultural land, this should conform to good quality criteria. So when we want to apply, okay, the municipal compost, okay, for agricultural land, this should conform to good quality criteria. The concentration of potentially toxic elements in some sewage sludge also must be accepted lower. Contamination can be unacceptably high. So thus, controls on discharge or sewers and treatment of sewage effluents to remove potential toxic elements should be actioned. So this is useful if we can do something to reduce the what they call it, the pollutants, the toxic elements in this for treatment for the sewage effluents. Furthermore, it is not recommended that municipal compost are mixed with sewage sludge, since this practice can increase the phytoavailability of potentially toxic elements. And finally, phytoextraction strategies can be employed to remediate contaminated land, and the plant material generated might be used for as biofuel. So these come with some ideas that we can use what they call it useful plants, okay, in on the mining side, on the municipal area or sewage or whatsoever, to make sure they can cleaning up the environment, and in the meantime they have the economic value. Okay, this is some of the plants useful for phytoremediation. I have some sort. Actually, I have many more pictures, but yeah, this is part of them. Okay, plants for phytoremediation. So this is the Ceres vetata, arsenic. This is one of the arsenic hyperaccumulator. Me and my friend, actually, Prof. Jane Yong from Sweden, we collaborate, okay, to come out with the to do the research. And I have my student also works on Ceres vetata, doing understanding the metal accumulation, arsenic or any other metal accumulation that can be absorbed or accumulated by this Ceres vetata. Okay, this Ceres vetata, this is from the family of Trudophyta, okay, family of fern, okay, and cultivated for ornamental, okay, can be cultivated for ornamental. Also, it's native and widespread in paleotropics. Okay, I believe in Indonesia also they have this kind of plants. Okay, but actually, this Ceres vetata, they can absorb very high concentration of arsenic. Okay. Very high concentration of arsenic, very useful. Okay, we can use this actually for phytoremediation, in cleaning up the arsenic, okay, for water and so on. And Melastoma malabatricum, this one is my research, my PhD research actually. My PhD research about the aluminum and ferrum accumulator. This is what I found from my research about the Melastoma malabatricum. In Malaysia, we call it as the senduduk, but I'm not sure about this in what you call it in Indonesia. What you call this? You might have your own what you call it vernacular name. And yes, this Melastoma malabatricum, they can accumulate very high concentration of aluminum, so they don't have any phytotoxicity symptoms. Okay, and it's a good model if we want to do about the aluminum accumulation study in plants. Okay, so habitat is a plant of secondary forest in the moist tropics, okay, in Southeast Asia. Okay, and that is why if we go outside, we can see at the roadside, okay, gap area, this plant will come first. This is actually among the pioneer plants in certain area. Why? Because they adaptable to the soil. When we try to plant, okay, crop planting the crops. They cannot survive with that kind of environment. The soil is lack of nutrient, okay, very high concentration of aluminum, but we can use this for the pioneer plants, okay, stabilizing the soils, which may create another ecosystem in the soil, the bacteria, the nutrient, and so on, which then might be useful for other plant species to grow. Okay, in some of the things, again, this Melastoma malabaticum, they have the what they call it, the medicinal value. The shoots and the roots are used to treat stomach and diarrhea. The roots and leaves are applied externally to wounds and hemorrhoids to hasten a recovery. So this is about the Melastoma malabaticum. When I did my study, my PhD study, I found that when we apply with certain aluminum treatment, aluminum concentration or aluminum addition on these plants, they absorb other metal elements, okay, very effectively. So when under the treatment, under the aluminum addition, so all the some of the metal elements also they can absorb, okay, arsenic, okay, manganese, they are all in the right direction, means that they have a positive significant correlation. When we apply this plant with aluminum, means that they have the potential to absorb other metal elements when they occurred at the right place and with high concentration of aluminum and so on. But this one, my students are doing this research. Well, me and our team, my students and so on, we are doing studying on how does the aluminum may affect melastoma, may enhance the Melastoma malabaricum in accumulating other metal elements. Okay, and we have Luigia octovalvis again. So this is one of the lead, zinc, copper, and arsenic accumulator. Okay, distribution-wide, distribution in tropical areas of many countries around the world. Habitat mostly in humid places. Again, use herbal medicine for treating various conditions, including edema, nephritis, and hypertension. Phytogramma colomelanos. Okay, 3DC. So habitat in crack of rocks. Okay, forest floor and full sun. Ornamental, it also quickly accumulates arsenic and has potential for use in cleansing soil and water contaminated by arsenic. So this is measuring the arsenic uptake by Phytogramma colomelanos, okay, by Young et al. 2010 from his paper. I got this from his paper. It's very interesting, very good research results, okay, where they have the potential of this plant, they can accumulate high concentration of arsenic. And other thing is Leperonia articulata. I believe in Indonesia we have this. Some of us in Malaysia, the indigenous people use this for making the straw. You know, they're making the straw from the Leperonia articulata. So it actually chromium nitrate survive in high aluminum and lead concentrations. So the habitat Leperonia articulata occurs in shallow water, usually less than 0.8 meter deep in open swampy locations, open marshes, swamps. These also have the potential for phytoremediation. Okay, in Borneo and South Sumatra, strongly completed closed Leperonia articulata mats have been used. Okay, so they use this for making a packing material for tobacco, rubber, kapur, cotton, and so on. And Scirpus grossus, sometimes people see this as a weed. Okay, habitat wetland. Okay, uses Scirpus grossus in Ayurvedic herb. So some of Indian in India, they use this for Ayurvedic herbs, used for the treatment for diarrhea, thirsty, nausea, scanty, and so on. So, but we have the phytoremediation wastewater containing lead. Solanum nigrum. So this is another plant that useful for phytoremediation. Common herb or short-lived perennial shrub found in many wooded areas, cultivated land, roadsides, and so on. Significant reduction level of cadmium in soil. So they have the potential in reducing the cadmium in soil. After 2 years of planting with same, the concentration in soil decreased. So what does it mean? They have this potential, but we are unaware about this. Okay, so actually they have this kind of this solanum actually came from the family of Chili, Solanaceae. Chrysopogons zizanoides, Poaceae. Okay, I believe in Indonesia also we have this, okay? You have this Crassopogons designer is widely distributed, okay? In Malaysia and Southeast Asia, potentially removing lead in the soil, okay? Habitat plant of the tropics, we found at elevation of up to 2,500 meters, okay? And it helps to stabilize the soil and protect it against erosion, but it can also protect field against pests and weeds, okay? From the roots, oil is extracted from and used for cosmetics, okay, and so on. They have certain useful elements, okay, and one of them is potentially removing lead in the soil, in the mining area. And yes, we have the buloh. The Phyllotaceous Persic. Okay, it is useful for landscape and soil erosion. But in the meantime, the buloh is very useful for the stabilizing the soil, and it was mentioned, it was reported by Björn et al. That some of the buloh, they are potentially accumulating certain metal elements such as copper, zinc, and cadmium. And in summary, okay, phytoremediation is an alternative way to remove contaminants from contaminated sources such as soil, water, and sediments by using plants. Okay, phytoremediation entails 5 processes of decontamination, such as rhizofiltration, phytostabilization, phytoextraction, phytovolatilization, and phytodegradation. These 5 pillars, okay, they have this kind of 5 process. Although phytoremediation is a promising method for alleviates metal in contaminated soil, but a proper management and selection of plants, okay, we need a proper management and selection of plants is required to ensure, okay, the excellent outcomes of this green technology. More research on phytoremediation is required, of course. Okay, more research on phytoremediation is required, especially in Indonesia, in Malaysia. Okay, we are, developing countries, that we need to ensure that our food crops is safe from this kind of metal elements. So the study on the pollutants, soil pollutants, okay, water pollutants is important. Yeah, and these are some of the reference I used. Okay, and that's all. Thank you.
Okay, thank you very much, Dr. Khairil. It's very interesting for your presentation addressing in this topic. Dear audience, if you have a question or comment, please raise your hand. And you can also write in the message or column chat.
Okay, maybe can I start with the question?
Oh yes, Bu Nunun, please.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Thank you, Dr. Khairil Mahmud, for nice presentation. Thank you, Pak. So nice and interesting for me, Dr. Khairil. So I have 2 questions actually. Maybe this is correlated with my interest too. So regarding to your presentation, do you have any measurement also for the gene expression and the metal transporter protein in that plant? And the second one, how did you have some approaches to enhance the metal transporter of gene expression to make the efficiency of the metal transport and uptake by those plants? Thank you. Thank you very much.
Thank you, Bu Nunun Barunawati.
Okay.
Yeah.
Thank you, Madam Nunun. Yeah.
Yeah.
So it's all about the metal transporter, okay, gene and so on. It's actually been reported by many previous research, okay, about the what they call it, the transporter. They involve certain plants. They have certain specific certain transporter that useful in transporting the what they call it, the metal elements. But in my study case, okay, I'm sorry to say that I do not cover the what they call it, the metal transport. my interest more looking on the physiological aspect, the aspect of how meta elements, they affect the photosynthesis rate, the plant growth, and then also the what we call it the relative growth rate and the nutrient concentrations. So from that is they actually, I agree with you, it's actually controlled by the gene. Okay, the meta transporter, or like I said, they have specialized. These plants are specialists in accumulating certain meta elements. I give you some example. Melastoma malabatricum, they are very specialized in accumulating aluminum. They have specific transporter. Yes, of course, they have specific transporter, and all these accumulation are very related with the gene. Okay, it is a special trait. When I did the phylogenetic trait, okay, we found that I did my research, what do you call it, my samplings at PASO research plot, CTFS plot in Negeri Sembilan, Malaysia. I believe while working with the what you call it, with the CTFS, they know these plots, 50 hectare plots, and we sampled the aluminum accumulator and non-aluminum accumulator. But at the end, according to the what you call it, when I did the analysis on the concentration of aluminum, we come out with the phylogenetic trait that we come out with the phylogenetic tree where we confirm that this is the phylogenetic trait. Plants accumulate meta elements, they have this is because of the phylogenetic trait. So it means that they are very related with the gene, with the transporter. So yeah, I mean I believe that that is actually one of my aim to study the metal transporter. Applying the grants for that, inshallah.
It's very interesting. Okay. Yeah, but don't know if you have feedback or.
No.
Oh no. Okay, thank you.
He did all of the intermediate plants that is also
Bununon, can you repeat it? We cannot hear clearly.
Oh, okay. Maybe the voice is up and down. Thank you because it's very nice. And thank you.
Okay. Yeah. Thank you, Bu, Mrs. Nunun. Thank you very much. Dr. Khairil, we also so many questions in the chat box. Okay, the first one is from Mochammad Faruqi, the Faculty of Agriculture, Universitas Brawijaya. My question is, phytoremediation already applied today, and did it have a negative effect for our environment?
Oh, phytoremediation has been may I see where is the question? It is in the chat box, right?
In the chat box, yeah. The first one.
The first one.
Sorry, I will. Oh, sorry. The first one is Moriana.
Oh, Moriana, yeah.
Please wait. It's so many, so I will check.
No, not Moriana. I think. Oh, Muhammad Al Faruqi. Okay, Al Faruqi. Yeah. Okay. Is it phytoremediation already applied today, and did it have a negative effect for our environment? No. I mean, it's not. For your information, there are many research being conducted about the phytoremediation, but they are more they're doing this kind of experiments in the lab. It is in the lab, not what they call it, not all research were conducted were applied in the field. Some of them yes, and they use some kind of plants in cleaning up the environment. Yes, I agree with that. But yeah, there are lots of potential of plant for phytoremediation. It's not negative effect to our environment, but actually they have a very good, effect in
Yeah.
cleaning up the environment.
Yeah, so it's a positive effect for environment to Mochammad Al-Faruqi. So it's important for us to apply this concept. However, we also have a question about how its remedial plant conserving strategy. This is a question for Ahmad Iqbal Maulana. I will repeat it. How it's a remedial plant conservation strategy to be applied in polluted river border area, especially in urban area. In several case studies in urban area, river borders are included in this area with densely population scale. For example, sorry, this is in Indonesia. In Surabaya is East Java also, and West Java and Jakarta.
Okay,
Thank you.
I understand the question. So how to apply the phytoremediation in such cases in a high polluted, a river or water bodies. Okay, so the issue is, yeah, we can apply it. No such thing we cannot apply it. So if you look, I like to put some. See my colleague, Prof. John is very expert in one of the phytoremediation. We have many discussion about this. So some of countries like Singapore, they develop some of what they call it, planting certain species in conserving the area, in avoiding in accumulating the what they call it, stabilizing that area from the metal contaminants. But I think in some cases it shouldn't be a problem at all. We can apply it. Okay, just find out, go and get some information about the what kind of metal elements in the soil or in the water. Come out, you can do some ex situ study, or in situ study, and then you can apply it. Shouldn't be a problem.
Okay. Yeah, I think it's clear for Ahmad Iqbal. You can plant it. What species? The specific for river border, Dr. Khairil.
Pardon?
What is the kind of plant, or which is the most species?
Okay. Just now you can see what they call it, the Lepironia. Okay. Some water bodies, water hygiene. Some of one of my colleague in Malaysia also found that the what they call it, some plants it is considered weed at the sawah.
Yeah.
They feel,
Yes,
but it is useful for removing the lead and plumbum in the water bodies.
Sometimes we're not sure that
we are unaware about that, but they have the benefits.
Yeah. Okay. And another question from Muhammad Romi Fikri. The question is, disadvantage of phytoremediation is cleaning up the environment. Is there any advantage from phytoremediation? It's similar like Mochammad Al-Faruqi.
Yes, disadvantage. I've been asked before with my colleague in the University of Putra, Malaysia.
Yeah.
They asked me, if you do the phytoremediation, what happened to the plants? Are you going to harvest and eat the plants? We are trying to not to eat the plants, but we can propose the right plants for economic value.
Okay.
Which have the value where we can use it for construction, example for buloh, for example, for plants, for especially in stabilizing the soil.
Okay. I think it's clear for Muhammad Romi Fikri. Thank you very much for your question. And another one from Keisha in India. It's also have a questions about the phytoremediation technique is one of the most effective technique that can help cleaning up the environment. Are there any disadvantages? It's also similar. I think
the technique. About the technique. I think Yeah,
It's
about
different.
the technique, actually we can, that's why I urge, I mean, I would like to, I mean, we have to do some, Indonesia, Malaysia collaboration maybe for phytoremediation studies, So actually we need to do lots of, studies about the phytoremediation to get planning or mitigation planning in cleaning up certain sites. That is, so about technique, yes, we have to discover more. Because some of my colleague, my Prof. John, actually, they have come out with the cleaning up the water bodies. They put the what they call teres vitata on the sea, on the water bodies, and they found that they can reduce the arsenic pollution or contaminants or compounds in the water. So means that they are actually moving to that level, okay, for the phytoremediation technique. About these advantages, it depends. We need to do lots of research about this. And currently, my student also doing my research about the phytoremediation, where we use certain plants.
Okay, Kenza, if you are very interesting to your topic research next, I think it's very interesting to you.
Come to Malaysia.
Yeah, come to Malaysia. Kenza, thank you very much for your question. And we have also a question from Halu Oleo University. For your information, Dr. Khairil, Halu Oleo is in Kendari, around Celebes Island or Sulawesi in Indonesia. Muhammad Taufik, according to your PowerPoint, bulo or bamboo in Indonesia is one of the phytoremediation plant species. Is there any experience related to researching the effectiveness of bamboo as a phytoremediation?
Researching the effectiveness of bamboo. Okay, so this is what my student now doing.
Oh, okay.
So this is what we are doing now, understanding the metal accumulation in bamboo.
Yeah.
Okay, because China, they also started with this, but we, Malaysia and Indonesia, we have lots of bamboo,
Yeah.
We have the bulo betung. I'm not sure what you call it, bulo betung, but we have the bulo lemang.
Yeah.
I think it is similar when the
Hari Raya,
similar. We eat the lemang,
Yeah. Salty with rice.
Satay with rice. We are actually similar, but then
yeah.
The bulo also, the name of the bulo, we call it as bulo lemang. Bulo lemang, and we have the bulo beting, bulo betung. Okay. So all these are actually native to our region.
Yeah.
So that is what I'm doing now. We are doing now on researching the potential of metal accumulation in bulo.
Yeah. Okay. Muhammad Taufik, thank you very much. If you have a question or comment, again, and very interesting topic, you can correspond with Dr. Khairil also. And to another question, Dian Novitri Rahmadani. Okay, my name is Dian. I want to ask about phytostabilization. When phytostabilization stabilizes soil contamination through compaction, deposition, and root absorption rather than cleaning, to do all three occur at the same times? Or when did it happen? Thank you.
On the root absorption rather than cleaning, do all three occur at the same time? It could be. Okay. When phytostabilization stabilize soil contamination through compaction, deposition, or root absorption rather than cleaning. Yeah, it depends on the species. Some species they not absorb it, but they have the what they call it mucilage. Okay, they have the cairan dalam akar tu yang boleh mereduce kan, boleh mengurangkan toxicity effect dalam akar. So indirectly they can change it to the less toxic forms. Okay, but some plants they change it, they stabilize and they absorb it. I can see Prof. Jean W.H. Yong to everyone. Okay, thanks Prof. John for coming. I invited him. He's one of my colleagues.
Yeah. Thank you very much for
and my mentor as well.
Okay. Okay, another question from Mochammad Joffrey. Okay, so it's mentioned that stabilization can stabilize soil by complexation and absorbs barot, and also some species can absorb contaminant element in their bodies. My question is, will the plant that absorb the contamination change the microbiological or physiological of the plant?
Okay, this is a good question.
Yeah.
Thanks, Joffrey. Okay, once they absorb, do they change? Do the morphological characters will change? How about the physiological? Will they change? No. Okay, in my case of study, when I did my research on the aluminium accumulation in melastoma in Senduduk, the physiological aspect change. Okay, with the aluminium, this plant then it stimulate the aluminium stimulate the uptake of other nutrients. Okay, it stimulates the uptake of other nutrients, means that they accumulate they increase the accumulation of nitrogen, phosphorus, and so on, compared to the melastoma without aluminum treatment. So this means that besides the photosynthesis rate also increase when we apply with aluminum compared to the plant without aluminum. What does it mean? It means that certain elements it trigger or stimulates to the increase in certain it benefits the plants, okay, physiologically. Okay, and how about the morphological? Yes, of course, they never change. It will never turn to another new species, but morphologically, it can increase the growth of the plants. It's similar to the tea. Okay, we have one staff from Brawijaya, I guess, Pak Wisnu.
Yeah,
his PhD. Okay, he's doing PhD with me. Okay, I am his supervisor. Okay, and Prof. John is a co-supervisor. So we are doing our research on tea. Tea actually accumulate high concentration of aluminum. Remember, guys. Remember all. The aluminum is toxic to most plants, but not to tea. So the tea, they can accumulate high concentration of aluminum. When under the what they call it, the presence of aluminum, it stimulates other nutrient uptakes. I mean, nitrogen, phosphorus, and this means that the aluminum itself is useful for the tea. The tea is good actually. They contain high concentration of aluminum, but bear in mind, okay, it's toxic. Okay, it's toxic. We are lucky. Why? Because our body system, we can tolerate to aluminum. If we cannot tolerate to aluminum, then we can get Alzheimer. You know, we cannot. Sometimes we forget our wife name. No, we don't know how to go back home. No, that's the problem sometimes. So, but we are tolerate to aluminum. Okay. So doesn't but actually tea is highly they have contain high antioxidant. Okay. So this is actually another useful thing. So it will change. The plants will become bigger. Okay. Physiologically, it helps the plants in stimulating other nutrient uptakes.
Okay, thank you very much.
Yeah.
So if you forget your wife, maybe so many accumulation in your body.
Tea is not dangerous.
That's dangerous. Okay,
you can drink it.
But yeah.
Okay.
Green tea or ocha is always we drink every day, so I think it's not dangerous. Yes, Dr. Khairil. Okay, another question from Haidar Fikri.
Okay, this is another good question.
Yeah.
So when will the plant be able to carry out the phytoremediation process? I mean, not carry out the phytor. When will the plant accumulate the better element? It is since the plant was planted or when it entered a certain phase. And does the plant have an age limit during the phytoremediation process? No. Okay. So actually, the plants they they can grow, they can survive. Okay. Like I said, melastoma in my case, we never apply with aluminum, but they can survive. But when we apply with aluminum, they are more happy. Okay, they are more happy. So it means that they will become bigger and Physiologically, they can stimulate other uptakes, okay, nutrient uptakes, and the photosynthesis rate will increase. Okay, do they have a age limit during the phytoremedia? No, I mean, no, they can still, but okay, the stages is different. The young shoot, in my cases of aluminum accumulation, the young shoot, the young leaves, they are more powerful, okay, in accumulating the metal elements compared to the old one, okay, because they are facing the development stages, okay. So the young tea, they contains higher concentration of aluminum compared to the young tea leaves, okay.
Yeah.
Okay, I'm talking about young tea leaves, okay. Because when we drink tea, I believe in Brawijaya, they have the tea, right? The tea plantation there. And if you go there, you go get the tea. Normally they use the young one, the young shoot.
Yeah.
They never use the old one.
The old one, yeah.
Yeah, but the young one contain higher concentration of aluminum and nutrients, and also the antioxidant compared to the old one. Okay, so similarly to other plants which we use for phytoremediation, and it's proven from my case of study in melastoma.
Okay, thank you very much. Haidar, if you have feedback or another question, please. And we're now going to another question from Erika, right? Erika question. Phytoremediation entails a 5 process of the contamination. If any of the 5 process are missed, can phytoremediation work optimally to reduce metal in contaminated soil and water?
No, no such thing like reduce metals in contaminated soil. Phytoremediation, not a 5 process. The 5 process involved in the what they call it in the phytoremediation. Yeah, it contain the phyto extraction and so on. But that all be considered as a phytoremediation. It can be either one. Okay, if any of the 5 process miss, no, yes, of course. If you can use it for stabilizing the soil, okay, it's considered as the phytoremediation as well. Okay.
Okay. Thank you very much. Any question or comment or enough? Okay, please, Dr. Khairil. Maybe another student will be right. However, it's very interesting, Dr. Khairil. So,
thank you.
So I have some question, but I think it's your experience.
Yeah.
How to dissemination your concept into community for us is dissemination the core concept to community services. Mostly we didn't care about the effect of the heavy metal. What's about this one? What's the experience about your experience?
Yeah, I mean, we are actually working on looking for a grant, for community grant, where we want to cleaning up the soils, okay, and stabilize the soils in Tasik Chini. It's one of the, if you Google the Tasik Chini, that is one of my research site when I did my master's. Okay, but as far just beside it, they happen what they call it the mining issue, some kind of mining, but not far. I mean, not big area, but yeah, but we are that place dominated by the indigenous people. You know, the orang asli. I don't know what Indonesia call.
Yes,
we call it as orang asli. Okay, they are the local and native. Okay. So they use the forest as their livelihood. Okay. So for the mining area, we actually wanted to plant useful plants for them for the economic value. In the meantime, we are trying to get the right plants to be planted there and stabilizing the soils and reducing the heavy metals there. So it actually helps the community, not about cleaning the sites, but also support them through the economic value.
Oh, okay.
Okay.
Thank you
So
very much.
that is we are working on that approach. Okay, we are working on that approach. Now universities all worldwide, they are talking about the community. So
Yeah.
Same to Malaysia. Yeah. So actually that is part of it, mining sites. But we can also propose some kind of integration, okay, between oil palm with some plants, okay, okay, with with some of the plants useful for phytoremediation.
Yeah.
So that is some kind of ideas that we need to dig out. We need to figure out okay, through the reading, through the experience, and also through the experiments and so on.
Okay, it's great. Thank you very much for Dr. Khairil or information for us.
Yeah.
And now we are going to the last section, certificate submission. Okay, thank you very much.
Oh, this is the...
Yeah, this is the certificate.
Okay. Thank you so much.
Okay, thank you very much for Dr. Khairil.
Yeah.
Okay. Thank you
So,
so much. This is the end of this section.
So
Dr.
I type
Yeah?
my email address. If any one of you want to get more information, you can
Okay,
just
sure.
email me.
Yeah, please.
Okay. So
Okay.
Khairil Mahmud at upm.edu. Thank you so much. Can we have a photo slot?
Yeah, sure. Okay. To Mrs. Paramita, please.
Okay, thank you so much for Dr. Khairil Mahmud for the great information. Then I will remember no cup of tea for my husband.
No, please give him the tea. No, we both our body will tolerate to this tea.
Yes, thank you so much. Very grateful presentation and very powerful session with Dr. Khairil Mahmud. And thank you so much also to Dr. Adistiawan for moderating this second session. So ladies and gentlemen, now we are going to the third session, which will be delivered by Mr. Raphael, Ph.D. And in the third session, Ms. Azeri Gautama Arifin, Master in Agriculture, will moderate. Ms. Azeri Gautama Arifin, Master in Agriculture, is lecturer in Department of Agronomy, Faculty of Agriculture, Universitas Brawijaya, Indonesia. And her educational background are Master in Agriculture at Master in Agriculture at University of Miyazaki, Japan, and Bachelor in Agriculture at Universitas Brawijaya. Her research interests in genetic transformation, genome editing, and also plant breeding. And if anybody want to contact with her, please contact in Azeri Gautama at ub.ac.id. Now to Ms. Azeri Gautama Arifin, please time and place are welcome.
Thank you, Ms. Paramita. Okay, I would also like to thank the next keynote speaker, Mr. Raphael Perez, and also all the audience for joining this third session of AMYCOS International Webinar. Okay, before I welcome Mr. Raphael Perez, I would like to read his short bio. Mr. Raphael Perez is from Montpellier, France. He got his Ph.D. in plant modeling and ecophysiology at Montpellier, France. He got his Master of Applied Science in Agronomy, Genetic, and Plant Breeding at NSAT. His Bachelor of Science in Biology, Chemistry, and Physics at Marcelin Papelotte. His research interests including plant modeling, ecophysiology, and plant breeding. And we got his email address here. So for anyone who wants to ask questions to Mr. Perez or maybe want to have a discussion, please jump to his email, raphael.perez@cirad.fr. So now, ladies and gentlemen, I would like to give a warmest welcome to Mr. Raphael Perez. Hello, Mr. Perez.
Hello.
How are you? Thank you.
Thank you for the invitation.
Joining us in this webinar. So, Mr. Perez, you have 45 minutes for presentations and 25 minutes for discussions. Okay, ladies and gentlemen, please welcome Mr. Raphael Perez.
Okay, thank you. So I will share my screen. I just need to find a good one. I think I will put it here. Okay, you will tell me if it's okay. Okay, do you have the full screen presentation or not? Maybe not. Is it okay or not? No, can you tell me? Not yet?
Not yet.
Okay, I will try to get, so maybe it's this one.
Okay, we can see it.
So do you have the full screen diapositive or not?
Yes.
Yes? Okay. Just I will. Okay. So can you see the point, the red point as well?
Yes.
Okay. So thank you for inviting me for this talk. So today I will present you the research I'm doing here in CIRAD since almost 9 years now. So I entered the CIRAD in 2011, and I worked on oil palm, and I'm still working on it. I've done some postdoctoral on corn, and since I start my research, I'm working on functional structural plant model, and so today I will present you different application on functional structural model of oil palm. Before this, I want to go back to the basis of agronomy and how plants grow. Because to model it, we need to understand how plants grow and product. So plants, to grow, they need resources, natural resources, like the sunlight to catch the light and use the carbon dioxide, and the water and minerals to grow. So here are the 4 main resources. But today I will only focus on the sunlight resources. And we'll see how we can model it and how we can use it for different applications in agronomy and basically in oil palm management. So how we can enhance plant productivity? So there is different factors. The first factor here is the environment. So the environment is how resources are available for the plant to grow. So it's all the climate variables, but also the water and so on. So we have no control. almost no control on light resource, for instance. But we can have some management practice to increase plant productivity that can be based on resource management, for instance, fertilizer or irrigation. But we can also work using cultural practices like pruning, leaf pruning in orchard, for instance, but it can be as well planting density. This is a management practice to increase crop productivity. And the last point, the last factor is the selection of the plant we will grow. So it's linked to breeding and to genetics. And what is important is to take into consideration the interaction between the environment, the genetic material, and the management practices, because it's the management of this interaction that helps us to improve production. So in the case of sunlight, the question I would like to address was how to enhance plant performance by optimizing light interception. So we have two factors mainly. We have plant breeding. That means we can select plants adapted to optimize light interception in a given environment. So mainly plant breeding will focus on plant architecture in the case of light interception. But also it can deal with management practices like planting density, for instance, or pruning in orchard and so on, different in trees in general. So if I come back to the history of breeding in rice, for instance, we realize that over the selection, the years of selection, we increased planting density in rice, and in the same time, by doing massal selection, we obtain plants, dwarf type plants, and new architecture of the plant that's enabled to tolerate higher densities. And afterwards, when we look at the architecture of the new plant type developed, we can see that their architecture is quite different, because they are smaller, they have leaf more erected. And so even if we didn't selected for architectural traits, we realized that how architecture was important in breeding program to improve productivity, and that the improvement of productivity also came from the increase in light interception. And it brings us to the concept of ideotype. So in the case of plant, ideotype is a type of plant that will maximize its performance in a given environment. And it was developed by Donald in 1968. So it's basically on this ideotype approach that we can work using modeling tool. So first, I would like to focus and try to address the question of how can plant models help to improve light interception efficiency. So first, I would like as well to come back to the base of models, and I will focus on Monti's equation, which tells us that the yield can be modeled as the quantity of light that is intercepted, that is available for the plants, and that is captured with a certain efficiency, what we called light interception efficiency here in red. This light energy will then be used with a certain efficiency to give carbon. This is the carbon assimilation efficiency through the process of photosynthesis. And lastly, we have another index, which is the harvest index, that tells us how all the biomass accumulated by the plant is allocated to the different part of the plant, and mainly to the part of the plant that is interesting for the yield. So basically fruits in fruit trees, or in grain in rice and so on. But it can be the whole biomass when we are valorizing when we want that the product, the output of the agronomic output is biomass. Okay, so here in this talk, I will only focus on the two efficiencies that are light interception and carbon assimilation efficiencies. I would like to present you as well the different models that we can use for different application in agronomy. We have what we call the crop models and what we call the functional structural plant models. And the difference between the two is a question of plant representation. In a crop, or also called process-based model, there is no representation of the plant, or partly, because we will only consider the leaf area of the plant, which is usually explicit, is state through the leaf area index. In a functional structural plant model, we have an explicit representation of the plant architecture with an architectural model which help us to have the three-dimensional structure of the plant and to simulate process on it. So here it's an example of 3D representation of corn. How we model light in this kind of model. So there is two different way of doing it according to the model we are working on. In crop model, light interception is based on what we call the Beer-Lambert law that derives from the physics, the physics. So light interception comes from incident light that will be intercepted with a certain efficiency. So in crop model, we worked with the fraction of light intercepted, which is an equation where we take into account the leaf area index. So there is no explicit representation of the plant, but an information of the leaf area per unit soil area. And there is another parameter, which is K, the extinction coefficient. So this is the way how we model light interception in crop model. And it is based on the assumption that the plant or the canopy is an homogeneous medium. And we can only simulate light interception at the plot scale. In a functional structural plant model, we have an explicit representation of the plant. We can simulate light on it using a light model. So the light model will simulate the position of the sunlight over the day, and we will simulate the sunbeam on the plant and where light is intercepted. So we can have an explicit representation and information of the quantity of light intercepted at the organ scale, at the leaves, that we can integrate at the plant scale and the plot scale. So actually, there is much more detail in a FSPM model compared to a crop model in terms of light interception. Then, when we have this light, we want biomass, because whole crop model are use biomass to model production afterwards. So there is two ways of doing it. In crop model, we will use another parameter called radiation use efficiency. So Montes showed lots of years ago that when integrated at the crop scale over a long period, a linear relationship appears between the total biomass and the cumulated intercepted light, as you can see here. And the radiation use efficiency is simply the slope of this relationship. Okay, so here you can see the slope can vary among the different spaces, but it's the same indices. In functional structural plant model, we will go into detail using biochemical approaches like the Farquhar model, for instance, that you may probably know. So here, this model we have an explicit estimation of light interception at the organ scale. We have the quantity of light intercepted, and so we can convert it with a certain efficiency according to biomechanical model. So here you have the response of carbon assimilation in function of photon flux density, so the light intercepted. So at the leaf scale, we have a nonlinear relationship due to the saturating effect of the light on photosynthesis, which can be accounted in a FSPM model, but is never accounted in crop model. So now I will focus. Now you know the difference between the model. And you know how we construct FSPM model. I will show you how we use FSPM model on oil palm and for different application. For instance, how we can assist plant phenotyping on complex trait like light interception efficiency. So here, this first part deals with what I have done during my PhD. And the question I addressed during my PhD was, how does oil palm architecture vary and how does this variability impact on light interception? So to make it simple, if we consider two plants, two genotypes, we wanted to know how the architecture impact light interception efficiency and afterwards plant performance. So to do it, we add a modeling approach and a field measurement strategy. So first, we wanted to evaluate architectural difference between progenies, because in oil palm, as we have lots of heterosis of the material, we worked with families of plants called progenies of siblings. So we wanted to evaluate if there is difference between progenies, to model them, to be sure that there is a genetic control of the architectural traits. And when we add the 3D mockups, 3D representation of each progenies, we use a light model to simulate light interception and to evaluate how genetic can affect light interception efficiency. So basically, from field measurement, we made an architectural model and we simulate light on it. So we couple an architectural model with a light interception model. So first, we need to define the architecture of palm tree, which is quite simple compared to other plants because there is no branching pattern. The oil palm follows the corner model, which is a monoaxial shoot with indeterminate growth and lateral inflorescences that can be either male or female. So when we want to model the architecture of a palm tree. Actually, we need to focus on the structure of the leaf because the architecture remains on the leaf and the structure of the leaf. Here I present you the leaf of a palm tree that is a rachis. Here you have the petiole is the first part of the rachis, and then you have the leaflets that are spaced along the rachis. As well, I wanted to present you how conventional density is organized in the palm tree plantation. It's a King Kong's design, so you have rows of palm tree that are planted in King Kongs. So you have the same distance between the whole plants. If we come back on the genetic control of architecture on other plants, there is lots of studies that have been done, and that show that, for instance, plant height is under genetic control. Branching pattern as well in trees are under genetic control, or even in rice, for instance, the number of tillers is controlled genetically. But also the leaf geometry. Here, for instance, you can see two leaves of different variety of tomato. You can see that they are completely different and they are controlled by genetic regions. In oil palm, there is a much lower number of studies. But some of them showed how the stem height can be controlled genetically. the leaf area as well, the vigor of the plant depends on the genetic material. And finally, the dimension of the leaflets also depend on the genetic of the material planted. So to develop the architectural model, we need architectural measurements or traits. So what we measured in the field, at the plant scale, we measured the height of the stem, the diameter of the stem, and how leaves are arranged. So what is called the phyllotaxy, so the angle between the consecutive leaves. At the leaf scale, and as I told you, the main architectural diversity remains on the leaf on palm tree. We needed to measure the length of the petiole, the length of the rachis, so the dimension of the rachis, but as well fine geometric attributes, such as the curvature of the leaf, the orientation of the leaf, and so on. And at the leaflet scale, we needed to measure as well the dimension of the leaflet and how they are organized along the rachis. So here I present you some results I published during my PhD and the difference between 5 progenies I studied. Here I show you, for instance, the ratio between the petiole and the rachis that can be significantly different between the progenies, as you can see. If we look at the length of the leaflets along the rachis, you can see that the trend is quite different between the different progenies, and we have a genetic control of the leaf length along the rachis. So we develop some allometric relationship to compare the genetic material and to be able to implement this allometries in the structural model. Here is a summary of all the traits that were with high heritability. So basically the dimension of the leaf and the leaflets were quite heritable between the genetic material. So here is, for instance, the output of our model when we integrated the architectural difference between the different genetic material we followed. I want to tell you as well that this study was done on young palm trees. They were not yet mature. So when we measure it over time, we can simulate the growth of the plant and the evolution of the architecture over time. When we have this 3D structure of the plant, we can simulate light on it, and we need a light model to do it. So what we have done was to make some replication to make virtual scene, so virtual plantation on which we will simulate the light, because we'll not simulate light on a plant alone, because it's not representative of what happens in the field. So we need to reproduce the same design that we have in the fields. So this pattern, and we have got lots of simulation to integrate the variability between progenies, but also the variability within progenies, because there is not a fixed material. All the plants are heterozygote, and you have some difference from one individual to another individual, even if you have the same progeny. So the light model used was one model developed here in CIRAD by Jean Doza, and it simulates the course of the sun during one day or during a longer period, and can simulate the different the radiation, the incident radiation over a day, over a longer period. To do it, we need to calibrate with field measurement, so climate data on radiation, to be sure to simulate the radiation that can be found in Indonesia, for instance, in our studied experiment. So here are some results of our simulation. So here you can see we simulate light on the 5 different progenies, and as you can see, so the color represents the irradiance, so it's the quantity of light intercepted per unit area. So as you can see, depending on the progenies, you can see that there is more or less light intercepted by the plant, or more or less light transmitted through on the soil. If we follow the fraction of light intercepted at the plot scale on the 5 different progenies, you can see that the cinetic is quite different, and you have one progeny with lower interception, mainly this one. And we have a significant difference if we look at a given time, the quantity of light intercepted at the plot scale between the progenies. So the efficiency of interception is different between the progenies. If we look at the quantity of light intercepted per unit of leaf area, you can see as well that it's quite different. And we were able to estimate that there is a significant effect of the progeny of light interception, and that 20% of the variability of light interception was not explained by leaf area. So it's not only the dimension of the plant that makes it to optimize light interception. It's also fine geometrical attribute, like the way leaves are erected or not, or the arrangement of leaf on the space. So the second question we wanted to address was, whoops, what happened? Where is my, sorry, there is a lag. Oh no. Sorry, my computer didn't like my presentation, I guess. I will need to, okay, sorry for this. I will, okay, it's coming back. I will go on the slide I was stopped. Okay, is it okay for you? It came back?
Yes.
Okay. Okay. So the second question we wanted to address was, okay, there is difference between progenies in terms of light interception, but which trait is interesting? Which trait optimize light interception for instance, for different application, like defining ideotypes or looking at specific traits for breeding? So we still use a modeling approach, and we use the coupling method between the architectural model and the light interception model. And so the idea was, we have an architectural model with inputs that are architectural variables, like the dimension of the leaf or the geometry of the leaf. So we have performed what we call the sensitivity analysis. So we change input parameters. So we change architectural feature, and we looked how this architectural change impact output variable, which is light interception. And so we can know which trait is really sensitive and explain the most how plants optimize light interception. To do this, we had a two-step approach. So the output we wanted to study was light interception efficiency at the plot scale, so the fraction of light intercepted, but also the carbon assimilation, the total carbon assimilated by the plant during a day. So the first method was a screening method. As we have an architectural model with lots of parameters, we had like 21 architectural parameters. We performed a screening method to see over time how architectural parameters affect light interception and carbon assimilation. So here, for instance, you have some representation of a given architecture. So one simulation where we selected randomly sets of architectural parameters. And as you can see, the shape and the dimension of the leaves are different between the different simulations. So when we add all these different architecture, we simulate light on it, and we estimate the efficiency to intercept light. And we were able to have a hierarchy of the importance of architectural traits. At the end of this study, we selected 9 architectural parameters that were the most significant parameters that explain light interception. These parameters are listed here. So it's the number of leaves, the length of the leaves, the structure of the leaf, how many leaflets on the leaf we have, but also a more tricky parameter like how the leaf is the bending of the leaf, what is the curvature of the leaf, and some parameter linked to the leaflet dimensions. So we took these 9 parameters and we performed a second sensitivity analysis, this time only on mature stand, with a method called the meta-modeling approach that help us to quantify the impact of each trait on light interception. And here I present you carbon assimilation. So first we wanted to be sure that our meta model, which is a model of the whole coupling method, so very simple model, that there is a good correlation between what was simulated using architectural model and light interception and what was predicted by the meta model. So as you can see, the prediction were very good. So we were confident on the output on the meta model, and we were able to investigate the behavior of the model. So using statistical approach, we were able to quantify the impact of each trait on carbon assimilation. In red, you have the principal effect. It means how one parameter affects carbon assimilation. And in yellow is the interaction between this parameter and all the other parameters. So as you can see, we have, for instance, the length of the rachis that is really important to optimize light interception and assimilation. And there is some interaction here. We can see as well that the number of leaf and the dimension of the leaflets were quite important as well. And finally, it helps us to define the plant that behaved, that maximized carbon assimilation. In the general architecture of those plants were erected leaves with short rachis and petiole, and with a high number of leaflets on the rachis. To go deeper, here I present you in these small plots is you have all the points. So one point is one simulation, one architecture that was done in this simulation study. And we have the relationship between carbon assimilation and leaf area index. As you can see, the more we have leaf area, the more we assimilate that light, but we have a saturating point. The first thing interesting is that the leaf area index, where the carbon assimilation was maximized, was the leaf area index we can actually find in the field. So even if we didn't consider actual architecture of the plant, because we have done sensitivity analysis, like simulating plenty of different architecture with some of them that are realist and some of them that are not realistic as well. We observe that the way to maximize light interception and carbon assimilation was on a range of leaf area index. That is what we obtain in the field. What we wanted to see was the difference between the plant that maximized assimilation at a given leaf area index and the one that had the lowest value of carbon assimilation for equivalent leaf area index. Here, if I represent, so in red, the plant that maximize, and in blue, the plant that minimize carbon assimilation, or leaf irradiance here, you can see that, interestingly, the plant that minimize carbon assimilation intercept more light at the top of the canopy, but much less light deeper in the canopy. In red, you can see that the interception of the first leaf and the top of the canopy was lower than the blue line, but after, when we go deeper into the canopy, we intercept more light. If we look at the carbon assimilation at the leaf scale, it's exactly the same. Actually, we have so much light here on the first leaf that we we overpassed the saturating point of carbon assimilation through photosynthesis. So actually the first leaf assimilate the same quantity of carbon, but when we go deeper on the canopy, you can see that we maximized carbon assimilation in lower in the canopy. So it's why those plants in red optimized carbon assimilation. Then in the second step, we wanted to see among all these plants that maximize carbon assimilation, what was the architecture of the plant. And we found 4 groups of plants, so 4 ideotypes with a specific combination of traits. Here there is a representation from leaflet to plot scale, and the color represents the carbon assimilation at the leaflet scale. So you can see that there is a trade-off between the size of the leaf and the curvature of the leaf. And we can propose 4 different ideotypes. What we observed as well is all these plants limit the mutual shading on the canopy. So there was no shade. We limit the shade from one plant to another in the field. This optimizes actually the light distribution within the canopy and afterward the carbon assimilation. So this part was more about the genetic control and how we can select traits to optimize light interception. But we can also, if you come back to the beginning of my presentation, as I said that there is one factor to control plant performance, that is the genetic, but there is also the management practice. So now I will go on another application, which is the use to use FSPM to investigate planting pattern and how planting pattern affect system performance in terms of light interception and carbon assimilation. So we are working with a company in Indonesia, PT Smart, you may know, and they were testing different design of planting pattern, either the conventional, the design A, and the design B, for instance, with a double density. We had 5 different designs that were with either difference in density or in the planting pattern, either in row or anyway, different pattern, as you can see here. We define a neighborhood index, which is the minimal distance between the plant. So here there is an homogeneous distance between the plant, but here, as you can see, within the row, you have a lower distance than between the row. So we use different distance and we looked at how this variable can affect plant architecture. I will come back on it. So how to investigate the effect of planting pattern on system performance? So first, we wanted to know how architecture is impacted by the design, because the plant can respond to the density. So for instance, if we take our row architectural model, I mean, in a given, in the conventional design, we have this architecture. We can simulate this architecture in this design and we have light interception and carbon assimilation. Now, if we look at the design with a higher density, we observe that the architecture of the plant is quite different. So we need to calibrate our model to take into account the plasticity of the plant in response to planting density and planting design. And we can simulate the mock-up in this design. We have a certain efficiency to intercept light. If we compare now the raw architecture without accounting for plasticity in the same design, and we compare the 2 outputs, we have the effect of plasticity on plant functioning. Now, if we compare in design B, the architecture of the plant in design B with design A with the architecture of plant in design A, we have the effect of design on plant functioning. So first, we looked at how plants respond to planting density. So here I present you, for instance, we use the neighborhood index, which is plant proximity, the distance between the plant, the minimal distance. So the color represents the minimal distance. Here, for instance, I show you the length of the rachis. If you look at the length of the rachis according to the leaf rank, that means the leaves from the top to the bottom of the plant, you can see that we have a very significant effect of the neighborhood index, so the plant proximity and the design on the length of the leaves. If we looked at the inclination as well of the leaves, we can see that there is an impact on planting density. We have leaves more erected when the density is higher. And we can integrate this plasticity pattern in our model, just adding a relationship between plasticity of a trait and neighborhood index. As you can see here, for instance, when the plant proximity is very close, we have longer leaves until a threshold where when the plants are very distant to each other, the leaf is quite small and stays small. For the declination, which is how the erectness of the plant, if you want, the closest the plant, the more erected the plant are. So we integrated all this architectural pattern in our model, and we were able to simulate the architecture of the plant in the different design. So here you have some output of the design. So the first result of this study was to know that plant proximity increased leaf dimension, leaf weight, and leaf erectness. We include it in the architectural model, as you can see, but we also find some architectural trait that didn't change with planting density. For instance, the frequency of the leaflet along the rachis is something that is really fixed by the genotype and is not controlled by or not altered, modulated by the environment. So we simulate light and carbon assimilation on the different 3D structure. And here I show you the difference between each design in terms of carbon assimilation at the plant scale. So at the plant scale, you can see that the plant that maximized carbon assimilation was in the reference design, in design A. On the other design, we have two colors because we wanted to know the effect of plasticity on light interception and carbon assimilation. So here in white is the difference between, it's the importance of plasticity in terms of carbon assimilation. You can see that it's quite not high, but for some design, there was the improvement of light interception due to plasticity and improvement of carbon assimilation. If we compare to the design A, which is the value in blue here, you can see that the carbon assimilation is much lower in all the other design than in the conventional design. This is because we have higher density in all the design compared to the control design, because we have less plant in the design A, so we have less competition for light, so more resources per plant. If now we look at the stand scale, so we take this value and we multiply it by the density, you can see that even if some design has exactly the same density, the difference in terms of efficiency to capture carbon is quite different between the design. Even, for instance, the design C has double the density compared to the conventional design, but there is no benefits because there is so many competition between plants that at the plot scale, there is no benefit. But we can see that some design, for instance, the design D, even if the density was increased, so there is much more plant per hectare, the efficiency to capture light and carbon assimilation was not so low compared to the conventional design. So when you increase the density, you increase as well the performance of the system. So this was really interesting. So we had like 40% more carbon assimilated in this design compared to the conventional design. So the go-home message of this study is architectural plasticity improve light interception via the expansion of leaf area. But the plasticity of the oil palm due to the monoaxial habit, there is no branching pattern. So it cannot optimize light in a way that the competition for light imposed by the design counterbalance the benefit of plasticity at the stunt scale. So we cannot optimize or increase plant performance only by putting more plants in a field. We need to think about how we plant, which pattern we need to adapt to maximize the plant. And so it's the idea of using functional structural plant model is we can make in silico or virtual experiment to see how one design or other design can be interesting. Because as you know, in oil palm industry, the cost to set up experiment is very high, and the time to get some result is very long as well. And the interest of modeling is really interesting in the case of oil palm. So this kind of approach, modeling approach, can also help us to define innovative design. For instance, we wanted to work in a new system integrating intercropping with oil palm. For instance, we have some loss of production in a monospecific system of oil palm. For instance, in the south of Sumatra, in Lampung, or in Palembang, for instance, you have El Niño effect with a very dry season sometimes, and that have a huge consequence on the plants because there is a water deficit and the production is going very low because the density actually and the competition for water is very high during El Niño. So the idea is maybe to reduce the planting density in a way that during the dry season there is lower competition between plants for the water. And during the rainy season, maybe we can think about planting rice, for instance, between palm trees. But this need in first to think about which design is the most adapted to this, which is the density that is interesting for have a good production in palm trees, but we need as well to have a system that allow enough light to be transmitted for the intercropping. So we used our modeling approach, and we make some change in the distance between trees and between rows of trees, and we simulated light on these different patterns. As you can see here, we test like eight patterns. So the density of the palm tree is changing, and here is the light transmission for rice. So as you can see, the lower if we have a lower number of palm trees on the plot, we have more light available for the rice. Here you have the relationship between the quantity of light transmitted for the rice and the palm density. So here is the red arrow is the conventional density. So as you can see, there is not so much transmission for the rice. But we can think that we can lower the number of palm trees per hectare in order to improve the light transmitted for the rice. And we find some interesting trade-off here. And this actually, this light regime simulated can be also an input for indoors experiment in controlled condition where we can modulate the light along the day. So we have performed one experiment here in CIRAD where we reproduce exactly what was simulated by our model in a specific pattern for intercropping, and we looked at the response of different varieties of rice to see if there is a way to improve, to do rice breeding for a given management practice with oil palm in intercropping. So this is something really interesting, and we are doing now, we are trying to do collaboration with Brawijaya University, mainly with Afifuddin Latif Adirejo. So here the idea is to, can we select rice for doing some intercropping with palm trees? So is there some tolerance to shade or to the fluctuation of light within a day? But one thing really interesting in modeling approach is to help us to better understand how plants behave, and mainly the response to climate change. Last year, we performed an experiment where we wanted to evaluate our modeling approach, because it's quite complicated to evaluate a model where you have a coupling between architecture, light model, photosynthesis, and so on. It's a combination of different models, but whole models are wrong. So when you couple them, you can have lots of bias from the different models. So we perform an experiment in very controlled conditions to be sure that we can evaluate the biophysical process, which is light interception and carbon assimilation on our plants and in our model. So we have done an experiment here in Montpellier. We have an ecotron platform, which is chamber where we can regulate everything: light, temperature, humidity, CO2, wind, everything is controlled. And we can measure the flux of CO2. So we had our plant, we make some measurement with LiDAR, with laser, and we reconstruct the actual 3D of the plant. In parallel, we measure using LICOR, maybe you know it, that enable us to estimate Farquhar parameters, for instance, so photosynthesis parameters and stomatal conductance model. And we observed within this structure the evolution of carbon dioxide and water consumption over a day. And we make some scenarios where, for instance, we modulate the quantity of CO2 on the chamber to simulate an increase in CO2 in the air. We change the radiation to have some cloudy days, sunny days. We change the temperature, the humidity, to see the impact of temperature on plant functioning, but also how vapor pressure deficit can change photosynthesis and so on. So it was really interesting because we had 4 plants, and we see how plant at the plant scale, all the flux of CO2 and transpiration. And as you can see, if you change the CO2, you will change the consumption of CO2 on water. And for instance, you can see that the CO2 flux were very low when we add very high temperature. So there is a response to the plant. And the idea is to evaluate our model. We will reproduce exactly the same condition on virtual plants and see how we are able to simulate exactly the same trends that we have here. So by doing this, if we have difference between our model and what we observed, it will help us to understand some processes that are not yet modeled in our approach, and maybe to find some clue about some processes that are not yet understood or ignored. To conclude about this presentation, I would like to present the whole approach we are doing now, and we are keep on doing this with our partner, Smart Research Institute in Sumatra. So the whole process is to simulate, to predict production according to climate, to genetic material, soil, planting design, and management. So we have a full integration of the process along time. So from young stage to mature stage, and in space as well, from leaflet scale to plot scale. And we want to test the effect of climate in the different process, either during the growth or at mature stage and so on. The effect as well of genotype and the effect of management practice. So it's the integration of all these processes that will help us to understand and to propose new system of production for oil palm. And so terima kasih, thank you for your attention, and I will be pleased to answer all your questions. Here are some references I used for this presentation.
Thank you, Mr. Raphael Perez. I need several important points from your presentations. You just mentioned that how important functional structural plant model is. So it can help assess plant phenotyping on complex traits like interceptions and carbon assimilations efficiency. It also can help to investigate the effect of architectural traits on plant performance and many more. So it's very interesting. Okay, ladies and gentlemen, now we move on the discussion sessions. So for anyone who want to ask questions to Mr. Raphael Perez, please raise your hand or write your questions in the chat box. Okay, so there is a question from Ms. Paramita. Has the plasticity of the architectural canopy been examined in relation to palm oil productions, and which factor is more important between density or cropping pattern, because the number of trees will affect oil productivity?
Yeah, thank you. It's a very good question. We have not yet the data about the production of the system, but it's really interesting because in our study, we saw how one design enabled to maximize or to optimize carbon assimilation, which was the design D, if you remember. We have not yet the production data, but we will have soon. And the idea is to see if optimizing carbon assimilation is enough to predict production. We know that there is lots of process after carbon assimilation, like carbon allocation, that can as well modulate production. So far, no, I have not yet the relation between production and plasticity. And the second question, actually, we couldn't decorrelate the density and the cropping pattern, because actually we could, because we have two designs with the same density, but the cropping pattern is different. And what was interesting is actually the cropping pattern is very, very important. Even if we have the same number of trees on the plot, the way they are arranged modulate light interception and competition for light. And actually, it's really important in terms of carbon assimilation. Now, in terms of productivity, we'll see when we will have the data. But very good question.
Thank you, Mr. Perez, for answering the questions. Ms. Paramita, if any feedback, please. Oh, okay. Okay, is there any questions from the participants? Okay, while waiting for another question, Mr. Perez, I have a question. So I'm curious about how to take data for cropping pattern, because I just saw the ecotron, but it's using camera. But how about the cropping pattern, or take a data to make a model picture? Yes, Is it
Yeah.
using manual or camera?
Yeah, actually, this study was not about the cropping pattern, but it's only at the plant scale. We want to simulate the performance at the plant scale. So the idea was we had a chamber where we have all the variables, the climate variables, and the flux of the plant. So the idea is to reproduce to reproduce exactly the same experiment, but in our computer. So we will make a 3D representation of the plant, but a 3D representation of the chamber as well. And we will simulate exactly the same light, and we will simulate the temperature. All the environmental factor will be the same in our simulation. And the idea is to simulate the flux of CO2 and the flux of water, and to compare it to what was measured. And we will see if our model is able to take into account the change in CO2 capture and in plant transpiration according to change in climate. But this is more, the objective is to evaluate the models and not the upscaling to the planting system. But now we are upscaling without being sure that the plant scale, the processes are well integrated. So it's why we wanted to perform this experiment, because we can have different formalism. Either, for instance, we consider so far that each leaf has the same efficiency to convert light into carbon. But maybe it's not the case. Maybe you have an effect of leaf age, of leaf position in the palm tree. So there is lots of assumptions so far that were not tested in our model. So it's why we have performed this experiment, to be sure that there is no to know or to limit the number of errors in our model approach.
Thank you. Thank you, Mr. Perez. It's very clear. Is there any questions from the participants? Okay, Mr. There is a question from Mr. Musokan Prasetyanto. Can we combine several species for one FSPM model?
Yes, actually, we will use two different models, one for alpine, for instance, and one for rice. So there is a different study doing this, and there is more and more with agroecological questions that are addressed nowadays. So it's already done. But the idea as well is to simplify sometimes the model, because when you mix plants, it means that you need lots of detail on different plants, and the simulation scene is quite high. So the computational demand is very high, and we are limited sometimes. So, for instance, there is some solution to combine a functional structural model, for instance, of the trees or the higher strata of the canopy, to have and then to use a crop model. For instance, we can think about using what I've done between rice and palm tree, and use only the output of the palm tree model to know the quantity of light or the climate condition below the tree, and to simulate with its input in a crop model of rice. So we can combine as well this approach. And I think there was one other question before this.
Yes, it's from There is a question from Adi Fajar. Is there any different or still fit with the model if we use the different or extremely different traits that used in the model?
I'm not sure to get the question. Actually, the trait, we didn't choose them because it depends on the biology of the plant. The model assumption or allometric relationship were choose to fit the best to the trait, to the observation. So for sure, if we change the formalism of the model, the result could be different. But what we wanted to do is to fit the most to the architecture. I hope I answered the question. Maybe I didn't catch it very well.
Yes, maybe Mr. Adi Fajar, if you have any feedback or you want to have a further discussions, please raise your questions. Okay. Okay, Mr. Perez, while we're waiting for another questions, I have one question. So our department is just built a new unit like plant modeling. But we don't know what to begin. So maybe you can give us some tips on where we should first do before we want to study about modeling.
Oh yeah, actually it will depend on the plant, and it depends as well as I present you. There is different way of modeling plants, and the question you want to address or the objective of your research will help you to select what kind of model you want to use. Usually when dealing with crops like rice or wheat, we use crop model. But it depends if it's for breeding, for instance. Nowadays, the functional structural model can be interesting because we go into detail. We can model traits that are actually measured in the field. And so, yeah, so begin is doing plant modeling. I think lots of things have been done so far. So you can, and the community of plant modeler is quite large, and they share what they are doing. So one advice is when you select the plant of study, first you look at everything that was already done on this plant, because codes are available, and there is lots of tool available on the worldwide. Maybe one thing is to call for partnership with people that already developed, and to adapt the model for the specific question you have. On which plant you will work? On rice or on, because here in my team we also work on rice or sorghum and oil palm.
Very informative, Mr. Perez. Thank you very much. Okay, there is one more question from Valdi Devalen. How to determine the right modeling for oil palm tree and slope area, and also keep constant to tree density?
It's a good question. So far, I have not tested the impact of slope area. You mean slope is? I guess it's when the soil is not flat. It's a good question, and it could be addressed with our modeling tool. We could simulate slope on the field and see how it impacts light interception, and for sure it will change the conventional planting pattern. Yeah. But it's good, very good question, and so far I have not worked on it. So if you are working on oil palm and you have some field with sloped area that we could get some data and compare to modeling, it would be very interesting. But for sure, there is probably the density of planting, or mainly the pattern of planting would be different in a slope area, because it depends on the orientation of the slope, if it's north, if it's west or east, because the course of the light is not the same. So for sure there is, and it's why now we are doing some computational experiment to see how to optimize planting pattern in regarding light interception.
Thank you, Mr. Perez. Is there any feedback, Mr. Ph.D? Oh, thank you. Thank you. Is there any question? I think there's no more question, Mr. Perez. So thank. Oh. Yeah.
Okay, so I need to take back.
Oh. Oh, it's from the first questions.
Yes.
From the question from Ade Budget.
Yeah, so to answer, actually, the genetic diversity of oil palm, of Elaeis guinensis, in terms of architecture is not so high. We will start to work on hybrid between Elaeis guinensis and oleifera, where the architecture is quite different. But so far, what we published in the last article is that actually the planting pattern has so much importance in light interception, in carbon assimilation, that, and the difference between genotype is not so high. So actually we will not have a big difference if we simulate other genotype. For instance, only to take into account plasticity or not showed us that there was no difference, or at least that the effect of planting pattern is so much important or higher than the effect of change in architecture of the plant, that to take into account plasticity or genotypic difference is negligible compared to planting density. So to answer the question, I think that even with extremely different genotype, but there is no extreme in palm tree, they are all close actually in terms of architecture, we will probably have similar results. Yes, I understood the question now. Thank you for the question.
Thank you, Mr. Perez and Mr. Ade Budget. Okay, ladies and gentlemen, this session is about to come to the very end. Okay, before I close the sessions, I will hand over the certificate as tribute to Mr. Perez. Thank you, Mr. Perez.
Thank you.
Please wait a minute. Please wait a moment. Certificate, please.
Thank you.
Okay, thank you, Mr. Perez. Okay, this is all for the third sessions. I will hand over to the MC. Thank you.
You're welcome. Thank you, Dr. Raphael Perez, for the great information about plant modeling, and also Ms. Ajari for moderating the third session. And ladies and gentlemen, now we have followed 3 great sessions today. Before we going to the next, we will have photo session. So please turn on your camera and give the best smile. Yes, please turn on your camera. Ready to take screen 1? Okay, already we're going to the second screen. Okay, continue to the third screen. Okay, is the last. Fourth screen, is there? Okay, already. Okay, thank you so much for all. Then we will going to the next session. Dr. Afifuddin Latif Adirejo, as Secretary in Department of Agronomy, will close this seminar. Dr. Afifuddin, please, time and place are yours.
Okay, thanks for giving me a couple of minutes, committee, especially Bu Paramita. Wow, first of all, I think it is very great situation and conditions. I observe since the morning up to the final sessions. I think all of presenters have presented their presentations in special condition for you all of audience. So thanks very much for all presenters, Dr. Raphael, Dr. Mochammad, Khairil Mochammad, and of course my colleague, Pak Roviq. So for you young learners, especially students, because here I think I observe also many students come here. This is not about theoretical point of view. This is not only about the data, okay? But this include the encourage. This is include the motivations. How can we use our information in the perspective of agroecotechnological point of view, for examples, and we can combine and we can compile together for our thinkings in our department in agronomy. So when you take your course, for example, especially our students, you can increase your capability. So this is what one of the message. And for our college, for our special college lecturer or maybe professor from our departments, thanks for participating this first meeting of international webinars. Yeah, and also we really appreciate all of the committee. You are the best, and the great commitment from you. Yeah, we really appreciate, because without you we cannot do this kind of great meeting, right? So and after this, after some remark that I have said before, yeah, we can conclude this meeting can reach, and this meeting has been accomplished or has been completely accomplished. Yeah, we give this remark to you, all of you here, for the committee, for the participant, and especially also for lecturer of Department of Agronomy. So give applause to you, all of you here. Yeah. And finally, thank you very much for assisting Department of Agronomy. Yeah, many presenters maybe want to share with us in further situations, but we can nice after that and we can discuss. I have some information for all students here that some scientists has contacted us. They would like to participate in international webinar like this, because the promotions from the committee can be seen outside from another scientist. So thanks you again for you, all of you here who have come to this seminar up to afternoon. Okay, and good day for you, every student, everybody here. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Afifuddin. We would like to thank for Mr. Mohammad Roviq, Mr. Khairil Mahmud, and also Mr. Raphael Perez as the speaker today, and also Mrs. Siska Fajriani, Mr. Adi Setiawan, and Ms. Azari Gautama Arifin as the moderator. And also big thanks for Dr. Nurrahmi Ardiarini and Dr. Afifuddin Latif, all lecturers and students of Universitas Brawijaya, the audience entirely, and also the committee. May what we have conducted will be useful for us. Amin. And ladies and gentlemen, if in guiding this event I made any mistakes, please forgive me. Thank you so much for your attendance, and see you again in the series of International Webinar of Agronomy Conference Series, or AMYCOS, from Department of Agronomy, Faculty of Agriculture, Universitas Brawijaya. Nice to meet you all. Goodbye and see you again.
See you. Thank you.